Yeah.
Good morning to all the dignitaries, guests, and participants. With great joy and immense exultation, I, Dr. Rekha Magirwar, feel privileged to extend a warm welcome for the celebration of World Fungus Day 2021. First of all, I would like to pay tribute to the great visionary, and our mission was spelled out long ago back, the founder president of Shri Shivaji Education Society, Dr. Punjab Rao Ali's Bhausaheb Deshmukh. Let us work hard, he had said, for the comprehension of scientific facts for the greatest good of the country. For many of the learned mycologists and distinguished researchers, faculties, and students from various places have joined for this celebration. The chairman for today's program, Dr. G.N. Chowdhury, Principal, Shri Shivaji Science College, Amravati, Chief guest and the inaugurator, Honorable Professor T.N. Lakhanpal, former President, MSI, and former Director, Himachal Pradesh University, Shimla. Professor B.F. Rodrigues, President of Mycological Society of India, Dr. D.J. Bhagiraj, and Professor N.S. Atri, former presidents of MSI, the Secretary of MSI, Professor N. Raman, resource persons, particularly Professor Rupam Kapoor, past President MSI and Head Department of Botany, University of Delhi, the office bearers and life members of MSI, principals, head of the departments, coordinators from collaborating institutes, students from various parts of India joined through the Zoom platform. It is a glorious moment. It's a 152nd birth anniversary of the father of the nation, Mahatma Gandhi ji. And the 2nd October also marks the birth anniversary of Lal Bahadur Shastri, Indian politician and the second Prime Minister of India. I extend my warm wishes on behalf of the organizers of World Fungus Day and Fungus Awareness Week 2021. Shri Shivaji Science College, Amravati is a leading institution imparting science education for graduation, post-graduation, and research degree under the aegis of Shri Shivaji Education Society. The Department of Botany has glorious history of having first recognized research lab for mycology in the region, and the noteworthy organization of National Conference on Mycodiversity with its sustainable exploration and biotechnological applications, MSEBA 2012, through its stakeholders. It's a proud to mention that in this conference, our department has conducted a session to address the problems of farmers, wherein the farmers directly interacted with renowned mycologists of our nation. Recently, we have conducted online national workshop on fungal systematics and technological advances 2020. As Honorable Line Body from UK has appealed all the mycologists to put on Fungus Day events, and they may be able to build up to a full launch of the World Fungus Day next year. So a little knowledge that acts is worth infinitely more than much knowledge that is idle. With this quote and background, our Shri Shivaji Science College has come up with the celebration of World Fungus Day today on 2nd October 2021 in collaboration with Mycological Society of India. I can proudly say that on this occasion, we have also conducted Fungus Awareness Week from 27th of September to 2nd October 2021 and received overpowering response of participants for all the competitions organized by 12 different institutions. We have experienced networking of highest order amongst them, and it is said that the mycelium is the neurological network of nature. With this, I would like to invite the organizing secretary of this event, Professor Dilip Hande, the council member of MSI, for his welcome address. Please, sir.
Good morning, ladies and gentlemen. It gives me immense pleasure to begin with this auspicious occasion by extending a hands of warm welcome for celebrating the World Fungus Day on 2nd October 2021. With due respect to founder president of Shri Shivaji Education Society, Amravati, Dr. Panjabra alias Bhau Sahib Deshmukh, and Goddess Saraswati. Today's day has a great meaning. On 2nd October every year, India celebrates the Mahatma Gandhi Jayanti in remembrance and honor of the birth of Mohandas Karamchand Gandhi and Lal Bahadur Shastri. So on this special occasion, I, Dr. Dilip Hande, organizing secretary of World Fungus Day celebration, heartily welcome you all for celebrating this momentous day. Honorable dignitaries on the dais, and who have joined through online platform, the chairman of today's function, Honorable Professor G.N. Chaudhary sir, Principal Sri Shivaji Science College, Amravati, Professor B.F. Rodrigues, President Mycological Society of India, Chief Guest Honorable Professor T.N. Lakhanpal, former President of MSI and former Director, Himachal Pradesh University, Shimla, Honorable past President of Mycological Society of India, Professor D.J. Bagiraj, Professor N.S. Satri, and Professor Rupam Kapoor, and many more senior mycologists. Most enthusiastic personality of the MSI, the Secretary, Professor N. Raman. It is a proud moment for us to have MSI office bearers, life members, organizing committee members, convener, principals, HODs from various colleges, students from various parts of the India. We have joined through the Zoom and YouTube platform. I extend my sincere thanks to MSI and its office bearer for giving us this great opportunity to host this first ever World Fungus Day 2021. I, on behalf of Sri Shivaji Science College and our parent society, Sri Shivaji Education Society, Amravati, wholeheartedly thank you for being with us. I appreciate and thank each and everyone who has put sincere efforts to make this event successful. It was a superb fungal awareness week celebrated from 27th September 2021 to 2nd October 2021. This fungus awareness program has a special importance and relevance in today's COVID-19 pandemic situation. I would like to specially mention the names of collaborating colleges, that is Brilal Biani Science College, Amravati, Sri Shivaji College of Art, Commerce and Science, Akola, Government Art and Commerce Girls College, Raipur, Vinayak Vignan Mahavidyalaya, Nangaon, Khandeshwar, Amravati, Bapurauji Butle College, Digras, Yavatmal, Sri Shivaji Art and Commerce Science College, Mutara, J.D. Patil Samrutta Mahavidyalaya, Daryapur, Rajeshri South Science College, Chandu Railway, SPM Science Gilani College, Ghatanji, Sri Shivaji Arts and Science College, Chikli, Sri Shivaji College of Agriculture Biotechnology, Amravati. As we all know, fungi are unique and large group of organisms found virtually in every ecological niche. The beauty and endless variety of fungi and mushroom enhance the biologists all over the world. About 2.7 million fungal species exist in the world, but only 1.2 million species have been described so far. Currently, there is a tremendous interest in the documentation of diversity of world's fungi. Biodiversity conservation are both defined on the basis of birds, mammals, and flowering plant diversity, but fungi don't get a look in. This means that habitat rich in fungal diversity are missed and remain unprotected. It is a well-known fact that fungi are prominent source of pharmaceutical and are utilized in several industrial fermentative processes, such as production of enzyme, vitamins, and pigments. Since last 50 years, many key advances in medicine come from lower organisms such as mold, yeast, and other diverse fungi. They are very helpful in producing high-value products like mycoproteins and act as a plant growth promoter and disease suppressors. Nowadays, fungal biotechnology is a key participant in global business due to its mind-blowing properties. Hence, after reviewing on the situation on fungal diversity and biotechnology, fungi are model research organism as well. But common people and even many biologists do not, however, realize the importance of fungi. So the awareness about this vital component of our planet must be spread throughout the world. If people know about something and care about it, there is a much stronger chance that they will protect it from the future of our ecosystem and indeed for the betterment of the mankind. With this context, after reviewing on the situation on fungal diversity, British Mycological Society has taken the initiative to organize World Fungus Day on 2nd October 2021 with the main aim to put on events for introducing fungi to general public. India is having a great legacy of mycologists and must get involved in this motto with British Mycological Society. To reciprocate the motto of British Mycological Society, Shri Shivaji Science College, Amravati and other colleges came forward to organize Fungal Awareness Week with renowned Mycological Society of India. As an organizing secretary of this program, I assure you that we will always be at the forefront to organize such enlightening events. We are obliged to MSI for showing the faith in our capabilities to carry out such a program. To conclude, I thank you all for your active and cooperative participation to make this event a grand success. I am also thankful to those who have attended this event online and encouraged us with their magnanimous presence. I hope to have a similar cooperation from all of you in future. Thank you. Thank you all. Have a good day.
Thank you so much, Dr. Dilip, for your enthusiastic welcome address. I thought a forest was made up of entirely of trees, but now I know that the foundation lies below ground in the fungi. So the vital and strong foundation lies below ground. With this quote, I now call upon Professor Vaishali Dange from Akola for introducing our guests. Please, Vaishali Madam.
Thank you, madam. Am I audible?
Yes, ma'am.
Yes.
Yes. Good morning to all. I, V.S. Patil, Professor Shri Shivaji College of Arts, Commerce and Science, Akola, feel extremely privileged and honored for the opportunity to welcome and introduce guests of Mycological Society of India on the occasion of World Fungus Day 2021. We are all celebrating Fungal Awareness Week starting from 27 September. Today is the birth anniversary of great leader Mahatma Gandhi and Lal Bahadur Shastri. We have gathered here not only to celebrate World Fungus Day organized by Shri Shivaji Science College, Amravati and MSI, but also for the guest lecture of eminent personality of Mycological Society of India. I feel proud to be a part of such an elite society where we have such a great personality whose work goes beyond India. We are grateful to all for accepting our invitation and becoming a guest of today's event. Our guest doesn't require any introduction. They are known for their high achievements of the institution. So it is such an honor for us to have you today between us. Professor Dr. N. Raman, Secretary of MSI, is Emeritus Professor at Center for Advanced Studies in Botany, University of Madras, Gundu Campus, Chennai. He was previously UGC BSR faculty, the Director of Center for Advanced Studies in Botany and also Director of Center of Herbal Sciences at University of Madras. He was also the Dean Examinations, University of Madras. He has contributed significantly on mycorrhizal biotechnology, herbal medicines, naturalized phytochemistry, and plant tissue culture. He has published 150 research articles in reputed national and international journals and 5 books. His books are referred by many researchers. He is the joint inventor of Austram, the herbal remedy for swine flu. He has developed a herbal drug for dengue. He has invented a new compound, mesoptine, which has anti-cancer as well as anti-diabetic effects, for which 2 patents have been granted. Professor Raman has guided 41 PhD students and 10 postdoctoral students. He has attended several conferences in many universities and delivered lectures in India and visited many countries. Professor Raman has carried out 25 research projects sponsored by UGC, DST, Department of Biotechnology, CSIR, ICAR, ICFR, Tamil Nadu Pollution Control Board, Tamil Nadu Forest Department, and industries such as Retort Pharmaceuticals and Sakti Masala. Professor Raman is accomplished with 22 awards such as UNESCO Young Scientist Award 1988, Bharat Ratna Sir M. Vishweshwaraya Memorial National Parisara Ratna Award 1999, Leading Scientist of the World Award 2005, Tamil Nadu Scientist Award 2006, Bharat Gaurav Award 2009, International Achievers Award 2009, the International Einstein Award for Scientific Achievements 2009, National Citizen Excellence Award 2009, Academic Achievement Award 2012, Mother Teresa Excellence Award 2012, Bharat Excellence Award 2014, Rashtriya Ratna Award 2014, Indian Award for Excellence in Education 2014, Hindustan Gaurav, Gagan Gaurav Award 2015. Our second eminent personality is Professor B.R. Rodrigues, President, MSI, is a senior professor in Department of Botany, University of Goa. He is the director of Internship, Incubation and Industry Partnership from 2018. Professor B.R. Rodrigues completed a dozen of major and minor research projects and attended over 165 national and international conferences, workshops, and seminars. He delivered plenary and lead lectures. He had chaired several scientific sessions. He had published 145 research papers in national and international reputed journals. He edited 5 books and guided 12 PhD students. Presently, he is guiding 5 students. He is a member of editorial board of many journals. He is a life member of Botanical Society of Goa, MSI, ISMPP, IMS, and INSOPP. He also acts as a reviewer for a number of national and international journals. He had got Young Scientist Research Project Award from DST, New Delhi, Excellence Award from Tanzanian Parent Network Foundation in 2010, Professor Bhide Memorial Award by Indian Society of Mycology and Plant Pathology in 2016, Best Teacher State Award from Government of Goa 2017, Dr. V. Agnihotri Memorial Award by Mycological Society of India in 2019. He is the Fellow of MSI, ISMPP, IMS, and President of MSI 2021 and 2022. Under his presidency of IIC, Goa University received 4 stars in 2018 to 19 and 4.5 stars in 2019 to 20 by HRRD New Delhi. He visited several countries. Our third eminent personality is none other than Professor T.N. Lakhanpal sir, chief guest of today's function, and also the former president of MSI, is a renowned meritorious and gold medal student in his college career. During his PhD study, he worked on cultivation of myxomycetes, which form the basis of a monograph on Indian myxomycetes published by J. Karmar in 1982. He joined Himachal Pradesh University, Shimla, and held till retirement in 2005. He served as a chairman of department and also dean of life sciences. He also served as a director, Institute of Integrated Himalayan Studies. After retirement, he was awarded Professor Emeritus by UGC. He also served as visiting professor in the Department of Forest Science, Oregon State University, USA, Mizoram University, Mizoram Department of Bioscience, Shri Sathya Sai University, Parasanthi, Nilayam, Andhra Pradesh. Dr. Lakhanpal has been a pioneer in research on cellular and acellular slime molds, mushrooms, and mycorrhiza. He has extensively explored the biodiversity of various groups of fungi from Northwest Himalaya, systematics, ecology, physiology, microbiology, cultivation, and mycorrhizal relationship. His technology has been utilized for tailoring seedlings of conifers and apple saplings for improved quality, quantity, and establishment at degraded sites. His research lab was recognized as a training center in mycorrhizal biotechnology by IDRC Canada and TERI New Delhi for financial support. He also carried out his work on Indian morels, that is morchilla. He developed technology for cultivation of shiitake and milky mushroom. Dr. Lakhanpal has served as the president of Indian Science Congress in Plant Sciences section, president of MSR, Indian Mushroom Growers Association. He also served as a member of QRT on Mushroom Biodiversity Authority of India, Biodiversity Board of Himachal Pradesh, DST, DBT, DOE, NF, GBPI, HEB, NRCM, UGC, and HFRI. He was chairman, RAC, director of Mushroom, Solent, chief editor of the Indian Journal of Mushroom and Indian Phytopathology, Indian Journal of Mycology and Plant Pathology, Journal of Tree Sciences, India, Journal of Microbiology, and Hill Journal of Botany. He also serves as recorder of Plant Sciences section of ISCA. He is a fellow of Indian Phytopathological Society and Society for Mycology and Plant Pathology. He was honored with the Best Teacher Award by Himachal Pradesh University, Shimla and Saraswati Award of Delhi University. He completed 10 major research projects of DST, DBT, DOE, ANAP, ICAR, GBPI, HED, and UGC. He delivered lectures in 65 universities and institutions across the country. Keeping in view his contribution in the field of mycology, 3 new species have been named after him. Dr. Lakhanpal has, to his credit, over 190 research papers, 55 contributed chapters, 5 monographs, 9 books, and 2 edited books. 30 students have received PhD under his supervision, visited countries like USA, Japan, England, and Thailand. So thank you very much. Over to Madam.
Thank you so much, Dr. Vaishali. Actually, we are having such an eminent personalities for today's inaugural session. These personalities don't need any introduction, and introducing them is just like carrying ice towards the Himalayas. So once again, thank you so much, Dr. Vaishali. Ladies and gentlemen, as you are aware that today's event is a collaborative event under the auspicious 2 major institutions. Number 1, Shri Shivaji Science College, and number 2, the Mycological Society of India. The MSI has been represented today by its Honorable President, Dr. Rodrigues from Goa University, and Secretary, Professor Raman, Career in Advanced Studies in Botany, University of Madras. We have been singularly fortunate in having Professor Raman with us for this mega event. Professor Raman is a task master at organizing such major academic ventures, and it appears to us that he has put all his accumulated experiences to the fullest use for this venture. We have heard the voice of the first organizer, that is Dr. Dilip Pandey. May I now invite Professor N. Raman, the Secretary of Mycological Society of India, for his welcome address. Please, sir.
Good morning to everybody. On behalf of Mycological Society of India, I welcome you all for this mega event, the World Fungal Day 2021. Professor Lynn Boddy from British Mycological Society has requested the MSI to have the World Fungus Day. So I think I am audible now. I am audible?
Yes.
Yeah, because some echo on both of my. So Professor Lynn Boddy from British Mycological Society of India, he requested the MSI to organize the World Fungus Day. So the Mycological Society of India has decided to conduct the World Fungus Day in various parts of India and various parts of India. And one of the places we selected is Shri Shivaji Science College at Amravati. Young and dynamic MSI member, Professor Ande, he came forward to organize this World Fungus Day in a bigger way. And apart from that, we requested the Annamalai University. There was a request from Annamalai University, Professor Kalachalvam. So the Amravati College, with the cooperation of the Amravati University and other colleges, they are making the World Fungus Week in an efficient way. So it made that the founder of the Shivaji College told that let us work together or let us work hard. But the founder of the Mycological Society of India, Professor C.V. Subramaniam, told that work together for success. So really the hard work and work together resulted in a mega event. And I welcome all of the MSI members and all other college participants and particularly Professor Lynn Body of British Mycological Society and other persons from abroad and India for this mega event, World Fungus Day. On behalf of Mycological Society of India, I wish that this function should be a successful one, and the sessions will be a good example for the young mycologist of India, a good example for them to organize such World Fungus Day in every corner of the India in the future. Thank you very much.
Thank you very much, sir. The credit for this overwhelming response lies with you, and we acknowledge your unstinted efforts. World Fungus Day is particularly significant in the content of specific area of mycology. Moreover, this event will add another feather in the crown of Shri Shivaji Science College. It was thrilling experience for all of us for Fungus Awareness Week. Let us have an expression of Head of the Department, Professor Dr. B.K. Dorkar. Please, sir.
Thank you, Rekha. Good morning, everyone. Chairman of today's function, Professor B.F. Rodrigues, President of Mycological Society of India. Honorable Principal Dr. G.N. Chaudhary, Shri Shivaji Science College, Amravati. Inaugurator, Honorable Professor T.N. Lakhanpal ji, former President of Mycological Society of India. Professor N. Raman, Secretary, Mycological Society of India. Organizing Secretary, Professor Dr. D.V. Hande, my colleague. Joint Organizing Secretary, Dr. Rekha Maggirwar. All teaching and non-teaching staff of the Department of Botany, Shri Shivaji Science College, Amravati. And all dignitaries joined through Zoom platform. I welcome you all to the celebration of First World Fungus Day. My botany department is established in 1958, is one of the leading departments in the field of teaching and research in plant sciences. Initially with BSc program, keeping pace with advances in various aspects of plant sciences, in 1977, the former principal Dr. V.R. Gurde instigated the proposal to get the recognition of research center to undertake the MPhil and PhD programs. Eventually, the PG programmer started in 2005, followed by COC program, that is Modern Horticulture and Landscape by UGC in 2006. Botany lab is the oldest and the first research lab of the college among the university. The department offers MSc Bioinformatics as an interdisciplinary course on self-finance basis since 2009. Since its inception, the progress of the department has been inextricably linked with efforts of former faculty members, to the name of you, Honorable Dr. V.R. Gurde, Mrs. Dr. Urmi Shah, Dr. P.R. Deshmukh, Dr. V.R. Deshmukh, Mrs. B.D. Konde and Professor P.W. Devtare. Even though the department has significantly contributed in various academic and research areas, it continues to strive for excellence. Currently, 8 faculty members out of 9 have acquired PhD degree and working on various research projects funded by UGC, DST, and other agencies. For about 5 decades after its establishment, the department is now recognized for teaching and research in the areas of mycology, fungal systematics and biotechnology, mycorrhizal biotechnology and plant pathology, ecology and taxonomy, plant physiology, water biology, and phytochemistry. Mycology laboratory was recognized by UGC for mycology research from 1978. I am happy to say that one of my colleagues, Professor Dr. Dilip Hande, is Shom Memorial Awardee. The extension activities of the department fulfill the need of society up to some extent through workshop on mushroom cultivation, vermicomposting, and environmental awareness, and protection through students' activities group EcoFriend. I wish a successful organization of the World Fungus Day 2021 ahead. Thank you very much.
Thank you so much, sir, for your this welcome address. It is a great pleasure for me to mention our college has brought many laurels, which is reflected thrice in the A grade conferred by NAC. The college has highly qualified, competent staff, well-equipped laboratories, and good results are due to the strong foundation laid by our senior faculty members. With this, I would like to now request Honorable Principal Dr. G.N. Chowdhury for his presidential address. Please, sir.
Hello, everyone. Good morning, one and all present here. First of all, I pay my tributes to Dr. Bhasai Punjab Deshmukh, founder president of Shri Shivaji Education Society, Amravati. Honorable President, Vice President, and all members of Mycological Society of India, invited speakers, Dr. Devtare, ex-head Department of Botany, convener, Professor Dr. Dorkar, head Department of Botany, organizing secretary, Dr. Hande, joint secretary, Dr. R.C. Maggirwar, Dr. Khedkar sir, scientist, research students, office bearers of Mycological Society, all teaching staff, organizing committee members, and dear students. It gives me great pleasure to welcome you all for this auspicious occasion of celebration World Fungus Day 2021. Shri Shivaji Science College Amravati is shouldering huge responsibility for upgrading the education system and always focusing on students' all-around development. Through vivid activities by our faculty members, we are also serving for the betterment of our society, mainly contributing the transfer of research from laboratory to land. It is pride moment for Shri Shivaji Science College Amravati and the organizer, Department of Botany, for celebration of World Fungus Day in collaboration with Mycological Society of India. As this department has organized many events with MSI, our bonding is very strong and we hope to share many more such events for the benefit of students and our society in future as well. For spreading knowledge for fungi, more than 12 colleges of different universities from Maharashtra and other states have organized several competitions, and I came to know that they have received huge response from the students. I would like to congratulate these colleges for their wholehearted involvement in their event and the winner as well. With such type of event, like World Fungus Day program can really achieve many goals. I believe that this program will prove itself a great motivation for scholars from different themes of mycological throughout the India. The Department of Botany deserves appreciation for organizing this program on a grand scale. My best wishes to the faculty, students, and all those who are associated with this program. Thank you very much.
Thank you so much, sir, for your motivational address, and thank you very much for your support and guidance for all such ventures. Organizing such a mega event is a golden opportunity, and the hosting department is pioneer and leader in the field of mycology. The joint venture with prime partner will definitely add more glory to the grand success of this celebration of World Fungus Day. May I now request Professor B.F. Rodrigues, President of Mycological Society of India, who has joined from Goa, for his presidential address. Please, sir.
Thank you. Good morning, everyone. The chief guest of this inaugural function, Professor T.N. Lakhanpal, other office bearers of Mycological Society of India, patron, Professor G.N. Chaudhary, Principal of Shivaji Science College, Amravati, convener, Dr. B.K. Dorkar, Professor and Head, Department of Botany, Shivaji Science College, Amravati, co-conveners, Organizing Secretary, Professor Dilip Hande, Joint Secretary, Dr. Rekha Magirwar, and other organizing committee members, eminent mycologists in this webinar, Professor D.J. Bhagyaraj, Professor E.N. Atri, and others, faculty members, researchers, and my student friends. Well, on behalf of Mycological Society of India, I welcome you all. At the outset, let me extend a very warm wishes on the occasion of Gandhi Jayanti and Lal Bahadur Shastri Jayanti to each and every one of you. And I hope that our efforts towards protection of our biodiversity that involves not only plants and animals, the fungi, the microorganisms, etc., along with the message of from our father of the nation about peace, would make this planet a better place to live in. Well, fungi are rightly the nurturing inhabitants of our planet. They are very important to mankind as mycorrhizal partners with plants. As saprophytes, they assist in nutrient recycling. As producers of food, medicines, dyes, etc. Besides, they are source of several enzymes which are used in industries. It is true that the absence of fungi from our ecosystem, absence of fungi, our ecosystems would fail to function. If you see the fossil records in mycorrhizal fungi, of course, I'll make my cut short my presidential address. Since I have a presentation a little later, but if you see the fossil records of EM fungi indicate that they were responsible for the evolution of land plants, and during the transition period way back 460 million years. Our country is endowed with high ecological diversity, ranging from alpine to tropical ecosystems, covering a total area of more than 3 million square kilometers, and has a rich fungal diversity. Of the 1/3 world fungal diversity existing in India, only 50% are characterized till date. Therefore, it is important to know about these microorganisms and to protect them. The Mycological Society of India is a scientific body founded in 1973 by a team of mycologists led by late Professor C.V. Subramanian, intended to bring together mycologists of the country and with the broad objective of promoting the development of mycology in our country. It is rightly said that it is better late than never. The celebrations of the first ever World Fungus Day is an initiative launched by the British Mycological Society, and MSI has rightly responded to their call. The celebration of World Fungus Day will go a long way in bringing awareness not only among the young mycologists, but also among the students who would take up researches in the field of mycology and in the common man. Well, it gives me an immense pleasure to know that Shivaji Science College, Amravati, in collaboration with MSI, is celebrating the first ever Fungus Day today as a part of the Fungus Awareness Week planned from 27th of September to today, that is 2nd October 2021. I believe that the various programs which are planned and which have been completed during this week-long celebrations would have an immense effect on the faculty, the students, as well as the general public. Also, side by side, the Annamala University has also initiated programs today to celebrate the World Fungus Day. I'm sure that these events will be, in future, these events will be celebrated with the involvement of many more institutions in our country, as well as abroad, and will have a great significance. We have made a humble beginning today, and this will go a long way. Well, I convey my best wishes to Professor Hon'ble and his team for the efforts that they have put in making this particular program or this particular awareness program a grand success. And I hope that this particular college has done many activities in the past, and I'm sure that they would be going ahead with many new programs in mycology in the coming years. Thank you for your patience. Thank you.
Thank you so much, sir, for your inspirational address. And as I'm working also in the field of mycorrhizal biotechnology, it is always a pleasure to listen to you, sir. Thank you once again for your inspirational words.
Okay, thanks.
This event, no doubt, will be a milestone on the forward journey of this college. We feel that our country should not lag behind in the forward march of science and technology. Mainly, our students must be able to compete globally, and they must get an opportunity to express themselves. On this special occasion, our organizing team have come up with the e-souvenir. This event will help to create awareness regarding the updates in the field of mycology. May I request the dignitaries on the dais to kindly release the souvenir and the technical experts to e-release the souvenir. Dear participants, this is the creation of souvenir committee. Please give a big round of applause for them. Thank you so much. Dear participants, it is an occasion like this. We get an opportunity to listen the stalwart personalities, and it's a great pleasure an honor to have Honorable Dr. T.N. Lakhanpal, former President, Mycological Society of India, as our chief guest and inaugurator for this mega celebration. I offer a very warm welcome to Professor Lakhanpal. He is a mycologist of international repute. We are excited to hear some words by our today's chief guest. So we kindly request respected Professor Lakhanpal to continue our event by sharing your words of wisdom with us and kindly declare the inauguration of this World Fungus Day 2021. Please, sir.
Thank you very much. And I welcome all the participants and grateful to the organizers for giving me this opportunity to interact with all. The World Fungus Day, as it is being celebrated today, is a very auspicious day because it falls on a day when our greatest leaders, Mahatma Gandhi and Lal Bahadur Shastri, were born on this day, and we all are indebted to them for the contribution they have made to the nation in the freedom struggle and for steering the country afterwards through hot waters. It was Lal Bahadur Shastri who gave a call to the nation for miss a meal so that we are self-reliant and we do not go out just for hunger with a begging bowl, and we become self-sufficient. And the same, it was the expression of the same words which the father of the nation had once said, that there is enough on this earth for the need of everybody, but there is not enough for the greed of everybody. So it was Lal Bahadur Shastri who implemented these words of the Mahatma. My heartiest congratulations to Dr. Dilip Pandey and his team, the principal, the head of the department, and other colleagues in the Department of Botany in Shri Shivaji Science College, Amravati. Really, when I think I visited, I attended 2 conferences there, and you deserve a long applause because you have carried forward the torch and illuminated the path for mycological research. The faculty and research facilities, they were added later. But the meager resources with which you initiated the work there, you really deserve heartiest congratulations. More so because you have been very consistent and regularly organizing conferences from day one and keeping everybody engaged. Now, thanks in no less measure to Mycological Society of India and especially to Professor Raman, who has been very, very encouraging, cooperative, and helpful in carrying out all the activities. In fact, left to me, I would not call him Secretary of the Mycological Society, but he is Secretary General of the Mycological Society of India, because all these years, how he has consistently carried out and worked for the upliftment of the society with so much zeal and a broad smile on his face that anybody he approaches cannot say no to him to become a member of the society. Fungi, all of us know, and they are only, the science of mycology is only hardly 300-year-old. But nevertheless, I think the fungi are the oldest of the organisms to have been known. Because when men learn to live on the bread and ferment and for alcoholic drinks, so the science of mycology started that day. And it continues to be the so till date. We have gone through many ups and downs. And at this juncture, the initiative that Professor Subramaniam had taken and all others had joined him and carried forward through this society, organized the mycologists, provided a platform, and a platform which has always worked for the upliftment of mycology. Dear friends, we have had a long journey, an arduous journey, many ups and downs, but with the cooperation and help of all of you and the new teams that have come up, the present president and Dr. Rupam Kapoor, Professor Atri for the journal, all these people, they have shouldered the responsibility to so nicely and so boldly that they could face hardships, still steer the ship through those hardships. And that is how we have come to this level. Heartiest congratulations to the organizations. Great applause to the Mycological Society of India, all the office bearers, past, present, and those who will join in future, and the stalwarts like Professor Bhagyaraj, Professor Manorachari, and all those who have been associated and carrying forward the cause of mycology. They all deserve great appreciation, and so do all of you. Thank you very much. Thank you very much. And I declare the World Fungus Day conference or the seminar open. Thank you.
Thank you so much, Honorable Professor T.N. Lakhanpal, sir, for your encouragement. And we will try to put your words into practice. And we are indebted to you for inaugurating this event. Thank you, sir. At the end of this inaugural session, I believe that the message we all must go home with is that the outcome of World Fungus Day should reflect as the community of concern, a community of stakeholders who have capacity to make a substantial difference to the way of use of mycology at every level. I hope you all are able to make great contribution to this process, dear participants. I would like to say that we don't scare easy. We are mycologists, and yes, fungi makes you brave. This is all for the student participants. On behalf of the organizing committee, I'm highly grateful to the chief guest, Professor T.N. Lakhanpal, sir, Professor B.K. Rodrigues, then the Secretary of MSI, Honorable Raman, sir, the chairman of today's inaugural session, Professor G.N. Chowdhury, our head of the department, Professor B.K. Dorkar, and all other dignitaries who have joined for this inaugural session and graced this occasion. So immediately we are going to start the first session, and may I now call upon Dr. Suruchi Kadu, Assistant Professor, Brijlal Biani Science College, to conduct the first session. Thank you. Thank you so much.
Thank you so much, ma'am. A very good morning, respected luminaire on the dais and off the dais, esteemed chief guest, all respected MSI members, and dear student participants. I am Suruchi Kadu, Assistant Professor, Department of Botany, Brijlal Biani Science College, Amravati. Welcome you all for the first technical session. For this session, we have respected Professor D.J. Bhagyaraj as a chairperson, Professor Navin Kango sir as a co-chairperson, and for this session, speaker is respected Professor B.F. Rodrigues sir. Before start the session, it's a great pleasure and honor for me to introduce today's chairperson and co-chairperson. Actually, Bhagyaraj sir and Navin Kango sir merely require any introduction, but still I try my best to explore some of their major achievements they have. Bhagyaraj sir has obtained MSc and PhD degree from University of Agriculture Science, Bangalore. He was UNDP Fellow of Australia and New Zealand. He was Fulbright Fellow of University of California. He also have NRAC Fellow of New Zealand, Commonwealth Science Fellow of UK. Sir has shown a great transformation in the field of mycology, though his unique and significant research work. His diligence based forward on his several prestigious awards. He was a fellow of Indian National Science Academy. He also a fellow of National Science Academy of Sciences and Agriculture Sciences of India. He is a fellow of Indian Pathological Society and Association of Microbiologists of India, Agriculture Biotechnology. He is a recipient of S.R. Vyas Memorial Award, Shom Memorial Award, Alumni Award from University of Agriculture Sciences, and he has Lifetime Achievement Award by NCOF Bangalore University. He is a recipient of Distinguished Asian Mycologist Award from Asian Mycological Association. Sir has published 413 research papers, research experience of about 50 years. He guided 40 MSc and 23 PhD students. For Sir's contribution in this field, he is honored with a new vamp fungi, which has been named as Glomus Bhagyaraji. This is a brief introduction about Dr. Bhagyaraj sir. I request sir to chair the session.
Thank you.
Next, our co-chairperson, Dr. Navin Kango sir. He is a professor and head of the Department of Microbiology and coordinator of Sophisticated Instrumentation Center of Dr. Hari Singh Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. He has more than 23 years of teaching and research experience. Sir did his MSc and PhD. He grabbed gold medal during his PG. He did his PhD in applied microbiology, biotechnology from Dr. Hari Singh Gaur Vishwavidyalaya. He did his postdoctorate from the Department of Biotechnology, Durban University of Technology, South Africa, and Department of Microbiology, University of Helsinki, Finland. He has published near about 45 papers, 11 book chapters, and 1 textbook of microbiology. He has completed 5 major projects. Sir guided 6 PhD students and 8 PG students. He is the fellow of Mycological Society of India and has several and he is a member of several professional bodies. With this brief introduction, I request Professor Naveen Kango sir to please co-chair the session. Now I hand over the mic to the chairperson. Over to you, sir.
Okay, thank you. Thank you very much. And I think we can go to the presentations. And first of all, I would like to thank the organizers for having given me this opportunity to chair this technical session number 1. And I also welcome the co-chairperson, Dr. Professor Naveen Kango, to this, and welcoming the Dr. Bernard Rodrigues, the first speaker in this particular session. And I think a brief introduction of Dr. Bernard has already been given in the inaugural session, but I will very briefly just give, because in case somebody missed that inaugural session and joined only now. So Professor Bernard Rodrigues is the senior professor at the Department of Botany, Goa University, and he is also president of the institution's Innovative Council, Goa University. His research area includes studies involving identification of novel AMP species, AMP fungal diversity in various habitats, taxonomy, inoculum production, and screening studies to identify efficient AMP species for various crop plants. Besides, he is also interested in use of AMP fungi in revegetation of mangroves, dunes, and mine wastes. He has completed dozen research projects and attended more than 165 national and international conferences, and has visited 8 different countries and published 145 research papers and edited 5 books. He has guided about 12 PhDs, and currently 5 students are working with him for their PhD degree. And he is the editorial board member of many journals and the life member of Botanical Society of Goa, MSI, Indian Mycological Society, and Indian Society of Plant Pathologists. And he has several awards to his credit. Just to mention a few, or the recipient of Young Scientist Research Award from DST, Excellence Award from Tanzanian Parents Network Foundation, Professor Bide Memorial Award by Indian Society of Mycology and Plant Pathology, Best Teacher State Award, Government of Goa, and Dr. V. Agnihotridu Memorial Award from MSI. And he is a fellow of MSI and Indian Society of Mycology and Plant Pathology and Indian Mycological Society. Currently, he is also president of the Mycological Society of India. Under his stewardship as president of institution's Innovative Council, Goa University was awarded 4.5 stars by the Ministry of Human Resources Development. And today he is going to talk to us on mycorrhizal fungi, a topic of interest to me as well as him and many others. Over to Dr. Bernard. Please unmute, Dr. Rodrigues. Unmute. Yeah.
Thank you, sir, for the nice introduction. Respected chairperson and my guru in mycorrhizal research, Professor DJ Bhagiraj, co-chairperson, Professor Navin Kengo, most revered mycologists, students, and friends. I think the slide is visible, my presentation. Is it visible?
Yes, sir. Yes,
Yeah,
sir.
it's visible, it's visible.
Yeah, thank you. So I have taken a topic that would benefit the students, especially the young students who are, I was told that there are nearly 2000 registrations, there are 2000 registrations, and these young researchers who would venture into mycology, I want many of them to get into mycorrhizal research, because that is one exciting area I find is important. And therefore I thought I would touch on the basic aspects of mycorrhizal fungi and also what we have been doing and how we go about working with mycorrhizal fungi. If you see, the fungi which are associated with the plants, there are different groups of fungi, like we have the coprophilous fungi, the endophytes, the lichens, which are in association, the fungi in association with the algae, then with the plant, various plant pathogens, disease-causing organisms, phyloplankton fungi, leta fungi, soil fungi, rhizosphere fungi. We have the mycorrhizal. The mycorrhizal fungi, they are an important group of fungi which benefits the plant growth, yield, and productivity. Now, coming to the historical of these mycorrhizal fungi or the fungi or the mycorrhizal fungi, it was Professor Francis Z. Kamienski who discovered the first the symbiotic association between the fungus and the root in Monotropa way back in 1881. He was a Polish mycologist, and later on Albert Bernard Frank, he coined the term mycorrhiza for the association in 1885. And at that time, people did not really believe that the mycorrhizal fungi would have a beneficial association with the plants. If you see the mycorrhizal system, it comprises of the plants, soil, and the mycorrhizal fungi. So you find that there are in the soil there is this whole web of mycorrhizal hyphae which are associated with the plants, and they provide nourishment or nutrient benefits to the plant. When it comes to the benefits, it's a mutually symbiotic association where there is bidirectional flux. That means both the partners are benefited. You find that the fungus, the plant gets nutrients, especially phosphorus and other nutrients like nitrogen and micronutrients from the fungus. From the fungus to the plant, and in return, the fungus gets. The plant provides carbohydrates to the fungus. Since these fungi, they are heterotrophic, therefore they depend on the plant for their carbon requirements. Now, I was telling in the morning during the inaugural that mycorrhiza played a very important role during the transition period, especially the transition from aquatic to land habitat, and that at that time, 460 million years back, it was very difficult. The conditions, the climatic conditions were different, totally different, because there were no land plants. At such a time, these fungi has assisted the plants to adapt themselves, to survive and adapt themselves in the terrestrial habitat. And if you see the research papers wherein they have identified the fossil mycorrhizal fungi way back in during the early Devonian period, they could get these orbiscal-like structures in the roots of plants, And also this is the vesicle, which is a fossil fungus, I mean fossil vesicle, which is recovered from the Ordovician period 460 million years back. And these are vesicles which are seen in the roots, again from the fossil specimen, thus indicating and probably proving that they have played a very, very important role. And if at all, I believe that if the human and the other animals have evolved on this planet, so also the plants is because of these mycorrhizal fungi. Now coming to the classification of mycorrhizal fungi, it is based on the classification based on infection anatomy. It's divided into two different classes, broad classes, the ectomycorrhizae and the endomycorrhizae. Now in the ectomycorrhizae, it is characterized by the presence of a fungal sheath which covers these roots, completely covers the roots. The sheath, when you take a section of these roots, then you can see the fungal sheath here, or the mantle we call it, and these fungi, they produce only intercellular colonization. You can see that the colonization here, and it forms a net-like structure which is called as the Hartig net. So the mantle and the Hartig net, these are two characteristic features by which you can identify whether the plant, I mean in the root, whether the plant is mycorrhizal, ectomycorrhizal or not. And besides, different fungi, they produce these colored roots because of the sheets, the mantles, the hyphae, they are variously colored. They produce such colorful patterns on the covering the root. Thereby, one can identify which species of ectomycorrhizal fungi is colonizing the particular plant. Coming to the endomycorrhizae, the second category, there are four different types in this. We have the arbutoid mycorrhizae or the ectendomycorrhizae. Now, this arbutoid mycorrhizae, they are characterized by the presence of these cruciform morphology. The roots show cruciform morphology, and that is, they have the hyphal mantle as well as you can see the Hartig net as well as the mantle. And besides, they have the colonization is both intercellular and intracellular. Therefore, it's called ectendomycorrhizae. It's a transitional stage between ectomycorrhizae and endomycorrhizae. And different species of fungi like Amanita, Cotinarius, Botrytis, different species of these genera, they cause the arbutoid mycorrhiza. Coming to the second type is the ericoid mycorrhiza, where it belongs, this type of colonization is seen in the family Ericaceae, where there is a ramification of each of the cell. So it forms a knot-like structure. You can see here about 80% of the volume is occupied by these coiled knots from the fungus, and that is the feature. And it's prevalent or predominant in the family Ericaceae, therefore it's called as ericoid mycorrhizae. The third category is the orchid mycorrhizae. The orchids are of ornamental value, and they are in great demand for their flowers. And the members of the genera like vanilla and epipegon and others, they are dependent on mycorrhiza for their growth. So the entry of the hyphae is a pre-requirement or prerequisite for further growth of these hyphae or for these orchids. And these orchids, when you take the section of the orchid roots, one can see the presence of these structures which are stained with trepan blue. These are called as the palatons. So when you see these palatons in orchids, then it's a confirmation that that particular plant is colonized by orchid mycorrhizae. So the species of Forms, Corticum, and Rhizoctonia, they cause orchid type of mycorrhizal colonization. And the last is the arbuscular mycorrhizae, or the AM fungi we call it. Earlier they were termed as VEM. The term, I mean, the alphabet V was deleted because all of these genera, they do not produce vesicles. They produce arbuscles, but they don't produce vesicles. Only a few genera they produce vesicles. Now coming to the different structures that are, how to know that a particular plant is mycorrhizal or is colonized by AM fungi. You can see that when you do the squashing of the roots by using the simple trepan blue staining technique, of Phillips and Heyman, you can see the presence of these hyphae which are aseptate. Only under certain conditions the septa may be formed. Like for example, in vitro conditions, we could encounter large amount of septation. But normally, in a normal condition, or possibly under extreme conditions, it could happen like the stress condition. But most of the time, you find that these hyphae are sinusitic, and then they produce these structures which are pastoria-like structures. These are called as the orbicles. Orbicles, they have an important role to play in the nutrient transfer. They transfer the nutrients across from the plant to the fungus and from the fungus to the plant. And this another feature of these orbicles is that they are formed as and when the nutrient requirement is there for the plant. And then after they are formed, after say a week's time, they would disintegrate. And you can see that they occupy the portion of the cell, of the cortical cells. So these they stain very beautifully, and then we have again the vesicles, another structure that is seen. The vesicles are structures which have dual functions. They act as chlamydospores, and with the return, they would be the resting spores during an unfavorable condition when the plant is, experiencing stress condition. Then the fungus starts producing this, these vesicles, and the storage, it stores the lipids, and with the return of the favorable condition, these they would act as chlamydospores, and they would function. They would colonize the roots of the plants. And in addition to that, they store the lipids. So they act as storage organs as well as the productive structures. Besides this, you have the spores, the intraradical and extraradical spores which are produced in the roots. Within the roots, the spores which are produced outside in the soil. So they produce this type of spores. And then the auxiliary cells. These are auxiliary cells in the roots, and these are auxiliary cells in the soil. So the auxiliary cells in the soils, they are colored in the roots. You can stain them with red and blue again. And they have structures like this. They may be finely accumulate or papillae, different type of structures based on which you can identify the species. It is possible to identify the species. Then all the genera, they don't produce these auxiliary cells. Only those that do not produce the vesicles are known to produce the auxiliary cells. And then we ventured into knowing that what these vesicles are. What these auxiliary cells are all about. And you can see that this is one type of auxiliary cell. This is another type of auxiliary cell where there is no papillae on this. And you can see that during the in vitro conditions, they produce the septa from septa. And then again, we try to stain them by using a Sudani blue stain, and found that it was staining indicating that they store the lipids. So this function of lipids was confirmed that they store lipids. Besides, we wanted to check whether they produce, whether they act as reproductive structures. But then, till date, we have not succeeded. We could see a structure like this. Probably we thought initially it is a germ tube, but later on we found that it may not. And we are working on this issue whether they can act as reproductive organs or structures or not. And coming to the benefits of these fungi. Now, why these fungi are important? One is there is no host specificity in mycorrhizal fungi, but there is something called host preference. That means a particular aim fungus would colonize the particular plant. Therefore, we need to screen and identify the efficient mycorrhizae for different crop plants so that we can increase the productivity. So when there is a host preference, then the amount of nutrients that are absorbed by the plants are higher, and therefore it will produce better yield. Besides this benefit, this is the reason why we have to actually start, one has to do mycorrhizal research. Other than that, the benefits include it increases the yield because of, one is that it helps the plant to, the plant fitness by getting optimum nutrients. Then increase water uptake, especially in plants of the dune, in the dunes, coastal dunes. You find that this is very important because the dune plants, they suffer for want of water, and therefore they are 10% of the plants we found that they were mycorrhizal. It maximizes the
all the
nutrient.
dignitaries, guests, and
uptake.
participants with great joy and immense exultation, I, Dr. Rekha Magirwar, feel privileged to extend a warm welcome.
Yeah. So it reduces the transplantation stress. Then when mycorrhizae help in the uptake of nutrients like phosphorus and nitrogen and other micronutrients, and you can reduce the fertilizer inputs, especially the phosphatic fertilizers, which are so very expensive because of the procedure that is involved in their preparations, and because of the fossil fuels that are used. Then it reduces the heavy metal toxicity. It is seen that in sites where there is heavy metal toxicity, these mycorrhizal fungi, they help the plant, they supplement in uptaking the heavy metals, in storing them in the vesicles and orbicals. It helps to maintain the soil fertility and disease resistance. A lot of work has been done by, Professor Jalali on the disease resistance by mycorrhizal fungi. Now coming to the how the mycorrhizae they help. This is the normal roots with the plant with the normal roots, and here we find that there is additional hyphae which are associated with the roots, and therefore the rhizosphere versus mycorrhizosphere. So when you have this mycorrhizae associated, they would uptake the nutrients where the roots cannot reach, where the normal roots cannot reach, and make it available to the plant. As a result, it has the effect on the shoot and the total biomass and yield. Here it is. You can see that this is the root. There is a depletion zone. Whatever the nutrients they get into this particular zone, they are easily absorbed by the plant roots. And phosphorus being a very less mobile nutrient in the soil, its movement is very sluggish. And for it to move from here and get into this zone, it takes a lot of time, and that limits the availability of phosphorus to the plants. And therefore, when you have the mycorrhizal association of the roots, would enhance or would enable the root to absorb more nutrients, because this hyphae would extend meters in the soil where the roots cannot reach, and they would absorb. Now, also the fine size of the hyphae would enable the hyphae to get into the finest crevices where available phosphorus is there in the soil and make it available to the plant. While the plant roots have limitations because of their size, you can see how these are the roots and these are the hyphae. They extend beyond the depletion zone and make the nutrients available. And you can see the difference in the growth. A simple experiment in Chile where there's a drastic difference in the growth when the mycorrhizal inoculum was added. Now, when we are working with the mycorrhizal research, then we need to work with the spores. So all the work with starts with the spores. Of course, we have to check the colonization of the roots, and then we need to isolate these fungal spores. There's a classical method of wet sieving and decanting by Gerdeman and Nicholson, which led to the increasing the pace of the mycorrhizal research, where is the most useful method till date, which is of course there have been modifications, additions to this. I would say there is sucrose density gradient method. These are the different sieves that are arranged in order, and then the soil is passed. The water, the supernet, and you have to take the soil along with the spores in the beaker, stir it, and after 2 minutes or so, then you have to pass it to the sieves, collect the sievings in the petri plates, and then observe it under the microscope so that you can see you'll be able to see a lot of soil particles, and then that you'll be able to see the spores. These spores we need to isolate. Once the spores are isolated, these are the isolated spores. These spores are of different sizes, different colors, ornamentation, and probably also of different ages. There could be spores in the soil for hundreds of years. There could be spores which are not viable. So we need to look at viable spores. If we take such spores and use it as an inoculum, it will not work. Therefore, we have to isolate the spores, and these spores, after isolation, they have to be trapped. We make this use of these trap cultures. So we try to trap the viable spores by using a suitable plant and then multiply the spores in this. There are other modifications, like we can directly use the sieving. You can use the entire plant and then place it in the soil, I mean in the pot along with this host plant. Then roots can be used, the rhizosphere soil can be used. So these are different modifications of the method. And then the selection of the host plant is important. Large number of host plants have been selected. We found that Coleus was a very interesting species, and then at present there are quite a few other species which have shown promise. And so these are the features of the host plant on which the selection has to be done. There should be more rooting. Here you find the roots are very soft, and you can easily do the wet, I mean the staining process, and see the colonization in the roots. They are better adapted, fast growing, readily they get colonized, and they are resistant to disease and pests. So we have used this as a very ideal host for not only for trap cultures but also for preparation of the or maintenance of the pure cultures. You can see the cultures are maintained in the polyhouse, and then of course it's a lot of work. We have about more than 25 species as on date which we are maintaining in the culture in the polyhouse. And some of the species like Cephalospora felinovi is novel species that we have isolated from this department. You can see the monocultures, how they appear. Yeah, these are the monocultures which are maintained in the, just take the phone. Monocultures. Am I audible, sir? Just a confirmation.
Yes, sir. Yes, sir. You are audible.
Okay. Thank you. Yeah. Then these are the pot cultures where we have monocultures, single spore cultures. When these monocultures, we periodically check them for colonization, whether they are colonized, they produce this type of arbuscles, the vesicles, and the spores. You can see the fresh spores, healthy spores. These healthy spores are very important as when you prepare the inoculum. This is one culture, Rhizoglomus irregularis, which we have identified, isolated. This can colonize the roots of plants within 21 days. So we are using it as an inoculum for one of the consortium that we are giving it for a consultancy work to one of the firms from Bangalore. And these are how the roots appear. They are full of these mycorrhizal spores, and these are extra radical spores. These are called as the intra-radical spores. Now, a few spore types, how the spores they differ. Like this is enterophosphoroid spore, which has it produces the secule. The mycelium would produce a secule, terminal secule like this, and this secule would store the nutrients. And then slowly there is development of a spore right at the base of the secule, and all the nutrients they are used for the developing spore. That's how the enterophosphoroid spore is formed. Whereas how it differentiates from the ethylesporoid spore is that here the spore is formed laterally. You can see the spore that is formed laterally. This is a secule, and that is the difference between both the types. And then we have the glomoid type of spore where it has the hypha is very predominant. In 99% of the cases, you will find the hypha attached to the spore, and you can easily identify that it is a glomoid spore or belonging to the glomus. And in echinospora, you find that most of the time the hypha would detach and the spores would be sessile. So these are some of the classifications. Classification because of the molecular work, it has there have been it has undergone a lot of changes. New families and you see the 2010 classification, 11 families and 18 genera of Schubler and Walker, and then Redeker 2013, it has 25 genera. So the number of genera have increased because of quite a few of these genera have arised, new genera which have single species. So which are marked with the asterisks. You can see Cephalospora is one where now they have 2 species, and therefore it's a strong case that it is a new genus with a new family. Coming to the different type of spores, spore identification is very important. You can see the Echinospora delicata, Echinospora delicata. These are delicata is a very delicate spore. When you just press it, it breaks, and then you can see such patterns on the spore. Echinospora levis, Echinospora reni, which shows very fine ornamentation. Then Ecolospora polonia, Ecolospora foveata. The foveata shows large warts on the spore, apical ornamentation in Ecolospora scorbicalata, where you can see the mature spore and the little younger spore, they show variation in the color. Then glomus, different species of glomus, glomerulatum multicole, which has got a large number of hyphal attachments. This glomerulatum has got two opposite attachments. Then some of the spores which are found as sporocarps.
Hello. Hello.
Then there is
Good
a
afternoon, Professor Rupam Kapoor.
fungal, the Pheniliformis, then Rhizophagus.
Yeah, it will be the next.
You can see the different ornamentations. Then this is Gygaspora, the diversity in Gygaspora, Gygaspora albita,
Okay.
Gygaspora renisporafora, Margarita. Again, if you see that there are hyphal appendages attached in addition to the bulbous suspensor in Gygaspora renisporafora. In Margarita, there are no hyphal appendages, and it is a distinct genus as compared to the Gygaspora. I mean, distinct species as compared to the Gygaspora margarita. When you have Gygaspora albida, Gygaspora margarita shows different the laminations. Again, in Recositra, earlier it was Scutellaspora, now it is the different genus show the presence of this germination shield. We'll come to that a little later. These are the sclerocystis, which are the sporocarpic forms. Sclerocystis cyanosa, Sclerocystis pakistanica, rediformis, Sclerospora. This is again Scutellaspora. You can see the germination shield here. They vary in color, ornamentation. They are very distinct ornamentation, like Scutellaspora nigra, which is a very dark colored spore as compared to Scutellaspora reticulata, which shows reticulate ornamentation. Then this is scanning electron micrograph of some of the mycorrhizal spores. This is Cephalospora felinovi, which shows very peculiar features on the scanning electron microscope. Now coming to the spore germination, there are two methods. One is the direct spore germination. Other one is the indirect spore germination. In direct spore germination, you find that the spore would germinate, it form a germ tube when you start germinating. So this is after 8 days. This is in case of glomus, Rhizoglomus sclerum, and this is after 12 days. Slowly the lateral hyphae would be formed, and then it would extend in the soil. Whereas the second type of germination is the indirect germination, like in case of Recositra verrucosa, you can see. This is the germination shield, which is found earlier, I said, in Gygaspora and Scutellaspora, in Scutellaspora species. And then you can see the presence of these germination shield. It has the germ tube initials from which the germ tubes would be extended. You can see the germ tube that is extended here, and this was that we collected directly from the soil, where we could see the germ tube. And here you can see this is in vitro, where there are a large number of germ tubes come. Since there are multiple germ tube initials, the multiple germ tubes are formed, and it is the indirect method. It's a better method in the sense that in those species, because it would initiate early colonization, faster growth of the hyphae. Then we have also these spore syndrome. Many times I find that the students, they are confused with this, and therefore this is basically to protect the spores. It is to protect the spores from the predators, and where the spores are produced in the older spores, spore cases, so that the predators would not damage or bruise the spores, so that otherwise the spore viability could be lost. This is Ecolospora soloidea. It's like the radiating sun's rays, and then this we have identified from our lab, Ecolospora soloidea. So also Cephalospora felinovi, the second species to Cephalospora baltica has been described from our lab, and you can see the distinct differences as compared to the Cephalospora baltica. Very close differences, but molecular sequencing has identified this as a distinct species very closely related to Cephalospora baltica. You can see that this type of, we need to do this type of taxonomical work so that we can differentiate. Like you can see here, in case of Baltica, you find that there are 4 outer layers, wall layers for the outer wall. This is spore wall 1. This is the second layer, the very fine, and this is the third layer. But in case of Cephalospora felinovi, there are 3 wall layers. Baltica has 4. Also, the third layer, the third wall of the outer layer, it shows birefringent properties, and it is much thicker. So also the spore wall 2, the second layer, it shows the thick. It's a thick wall, which is something like much thicker than the Cephalospora baltica. So 1.3 to 1.8 fold thicker than Cephalospora baltica. Also, we have the warts on the Cephalospora felinovi. They are much denser, 3 to 4 times more dense than in case of Baltica. And they also show the hyphal mantle. It shows more or less similar features, but you can see those of Cephalospora felinovi. They are straight and they have numerous branches at inclination relative to the parent hypha. Whereas in Baltica, they are generally sinus. So these characteristics have helped us to identify that it is a different species. This is again where we could finally get the orbicals. We could stain the orbicals and observe them, and say that it is a Cephalospora felinovi. You can see the felinovi is Cephalospora felinovi is very distinctly related to Baltica, and they fall in the same. There are other species which are unidentified, which are being identified. You can see how the species produce multiple sacules, and so that the spore is formed. So also there are other species which shows a very definite ornamentation pattern. We have also done work on Pisolitis albus, which is ectomycorrhizal fungus, where we have been able to grow it in culture. This fungus was collected from Acacia auriculaformis from the registry, and then from the leaf litter, and we could develop the inoculum. The culture, and this is the pelletization, where we could get pellets, and we also tried growing the pellets on the solid medium, and solid pellet on growing on the solid medium, and this is a normal mycelium, and we try to see whether there is somatic compatibility in that. These are the spores. So this particular inoculum, with the help of a carrier, is used for mycorrhization of the plants in the stress conditions. Now coming to a little to work on the in vitro cultivation, we have been able to grow these mycorrhizal fungi in the culture by using the minimal monogenic minimal medium of Becardin 14 and the modified Roman medium Diclage. We have tried to modify the medium, and it was working well for our cultures. This is the root organ culture, and then you can see the different spore germinations. These are from the spores from the pure cultures. You can see that very clear and fresh spores that we could isolate from the pure cultures. You can see the transform route that we have been using for colonizing and then in the petri plates, there's good amount of colonization and the hyphal growth, and you can see the spores, the sporulation in different species, correlation in different species. Gygospora disciplines, you can see the colonization taking place here, and the sporulation, young spores that are formed in culture. Then this is in Funiliformis mossae, where there is sporulation. This is in Scobiculata. We have a dozen of species like that. The Rhizoglomus sclerum, which could produce about 25,000 spores in a single petri plate. And then we also use the vesicles as propagules, and we could generate very good sporulation by using vesicles as the starting materials. Then coming to the last part of this presentation is we tried working. The whole idea is to identify the efficient species for different crop plants. We have tried a number of plant species. There's a lot of data generated, but I've taken only a few examples to show that Claridodermus etunicatum has been found effective for in case of chili, where it increased the yield. This is in case of okra, the bindi, where that is esculenta, where it is where the scobiculata, Eccleospora scobiculata could show the better host preference. In case of Perica papaya, we found Glomus mosaic as the most important fungus that could be used, efficient fungus that could be used to increase the mineral nutrition as well as the yield in case of papaya. We worked on the medicinal plants like Amravati paniculata, where Tigaspora albida was found to be the better, which it also resulted in the increase in the secondary metabolite, that is endographoloid, which is a diterpenoid, which has medicinal values. Then coming to the work in the ornamentals, like in case of chrysanthemum, there was not only increase in the number of flowering, but the days of flowering, the number of days of flowering were reduced in mycorrhizal plants. Then in the control, this is in case of Crasendra infundiliformis, where we found Rhizoglomus intradices was a better candidate for colonizing. Some of the mangrove plant species, we found Glomus sclerum as the good species that would increase the growth in case of Cereops segal, a mangrove plant species. Not only the growth in the shoot, but also it increased the growth in the roots, because in mycorrhization the root growth is very, very slow. And then this is in case of uninoculated control. This is in case of control. We find that we could increase the growth, significant increase in growth of the roots as well as increase in the shoot. So this increase in growth increased the amount of colonization, and therefore better uptake in this mangrove plant species. We have also been working with the reclamation of the minelands or revegetation of the minelands, where we have identified mycorrhizal species. Efficient mycorrhizal species are different for different plants that we use for revegetation of the minelands, planted them on the dunes, and we could definitely see that these plants could survive under the stressful conditions. The survival percentage was quite high, about 80% of the plants survive, which mycorrhizal plants survive on the dumps. Where during the months of, say, right from February to May, there's hardly any water. There was a heavy water scarcity in these areas, but still the plants survive. Now the inoculum that we produced, the pot culture inoculum, the in vitro inoculum, we try to develop a carrier for this, and the different amendments were done, different material substrate were mixed as carrier, and we found that the combination of different substrate makes a difference. And we found that we could get 98.2% colonization in this six number substrate that we have developed. This is we have used Elysium coricana or the finger millet as a host plant, and this is Rhizoglomus or Rhizophagus intradices was used as the fungal. Then the development, the carrier is developed and it is multiplied in trays. And the coleus plant that we inoculated with the monogenically produced cultures. You can see the distinct differences in growth of the coleus plants. Very healthy plants. When we checked for the root colonization, it showed, very high amount of root colonization in the roots. Also, it produces the vesicles, orbicles, and also hyphae. This was in case of where we used the in vitro produced inoculum in case of Anacardium occidental, which is a plant that we use both for the mine-based reclamation as well as for the dune reclamation. So it showed differences in growth. Rhizoglomus intradices again proved to be a better candidate for the Anacardium plants. And also you could see the amount of colonization, the formation of orbicles and vesicles and the spores. They were, compared to control, we produce a large number of these, indicating that this particular fungus, it can work efficient in the Anacardium plant, and therefore this colonization of these plants or initial colonization of the mycorrhizal, the Anacardium plant with efficient mycorrhizal inoculum would help better growth and survival either on the dunes or in the mines. So therefore, in the end, I would like to conclude saying that when we work with mycorrhiza, then we have to take the different aspects into consideration, right from isolation, right from checking the colonization, whether the plant is colonized or not, up to the isolation of the isolation of the spore, and then the proper identification of the spore trapping of the trap culture, I mean, preparation of trap cultures is very, very important thing. Otherwise, most of the students, they send me slides where the spores are, you find that they are either parasitized or they are very difficult to identify. So therefore, when you prepare the trap cultures, then you get only the ones which are viable, and prepare the trap cultures 3 times, 3 life cycles, so that whatever any point of time any species is dormant, we find that that species would be after completing its dormancy would colonize. So that's how it's not that we identify the spore diversity just by once, but it is done 3 times or 3 cycles, so that we can know the viable propagules in the rhizosphere of those plant species. Therefore, we can assess the diversity properly, correctly, and that would help. And of course, preparation of the inoculum, you can prepare the in vitro inoculum, or you can simply prepare the pot cultures or the pot culture inoculum. That also works very well, but you have to think out of box so that how to prepare this pot culture. Many times people talk about contamination and other things, the endophytes, but we need to handle this very cleverly. That is what is important. That is what has gone through, and we have already initiated a pot culture inoculum, and we have a lot of commercial entrepreneurs who are coming to us asking for this. Right now, we have a consultancy to one of the firms who are working on coconut to identify the fungal and to produce the inoculum on coconut. So that is very, very important. And then that completes your work rather than simply doing diversity or colonization. So with these few words, I would like to thank the college, Professor Dilip Pandey, for giving me this privilege to make this small presentation. And I'm sure that the students would take it in a big way. And many of the students who are present today and hearing this particular presentation would venture into mycorrhizal research and do many great things than what we have done so far in mycorrhizal research. Thank you.
Thank you very much, Dr. Bernard. So I think we are about 10 minutes behind the schedule. And so if there are 1 or 2 quick questions only, probably rest of it you can ask, probably contact Dr. Bernard later. If 1 or 2 quick questions, please. Yeah, Dr. Rupam.
Good afternoon to all, and Professor Rodrigues. It was a delight listening to you. We are still struggling with the culture of fungi. So every time we do an experiment, we have to procure a fresh lot of inoculum. When you said 3 cycles of trap culture, is it how long each cycle would be? And do you advise changing the host every time?
Yeah, ma'am. The each cycle would be for about 4 months. So 90 days and plus drying and all would take around. Then you identify the species. You know the fresh spores from the old spores. One. Secondly, when you start with the first cycle, you check the colonization at the end of say 30 days or 35 days, and you are sure that the root is getting colonized. Otherwise, if all the spores are dormant or they are not viable, then you never get colonization. So once that is done, the first cycle completes. Again, you isolate it and see which spores have increased in number. And what was the second question, ma'am?
Do you recommend changing the host every time we start a cycle?
Yes. One can always try more number of hosts. Maybe 3-4 hosts you can try, and then you can narrow it down to 1 or 2 hosts. When you find that the host is giving you the desired result, no issues. But you can always, that's what we are doing. We have a little problem with coleus, like in the summer months, it is very hot outside, and then these coleus plants, they find it difficult to withstand the condition. Therefore, that we use Eleusine coricana, that is the finger millets. In the months of say March, April, May, that is the time we use that, and we use a combination of this both as well, and it is working very nicely.
Okay, thank you. And then probably because of the time constraint, we'll have 1 more question. That's it. You can contact, I think, Dr. Bernard also later. I'm sure that he'll be willing to answer you. Am I right, Dr. Bernard?
Yes, sir.
Okay, I request Dr. Kango, just a few words in conclusion. Unmute, please.
I'm not able, yeah. Thank you, Professor Bhagiraj. And it was a pleasure to listen to Professor Rodrigues, President Mycological Society of India. And he has given an elaborate account of mycorrhizal fungi, their characteristics and their applications. I'm sure all of us, especially youngsters, must be happy today to see wonderful slides, wonderful pictures, and the kind of applications you can do with mycorrhizae. So I'm very happy that on the very first World Fungus Day, we could witness the beautiful morphology of the wonderful mycorrhizae. So I won't take long. Thank you so much for allowing me to join this session. And it was wonderful to be with you, sir. Thank you.
Thank you very much. And I'd like to thank the organizers once again for giving us this opportunity. Hand it over to the organizers. Dr. Dilip Pandey.
Thank you. Thank you, sir, for chairing the session, and the co-chair, Dr. Naveen Kango. Professor Andai.
Thank you, sir.
Thank you so much, sir. That was a really remarkable session. Thank you so much. Sir, your words enlighten us and student participants. Now, I hand over mic to the Dr. Kishore Surarkar for the vote of thanks.
Vote of thanks.
A very good afternoon, one and all, respected mycologists present here. Myself, Dr. Kishore Suradkar. Today we are being celebrating the World Fungus Day 2021. We have just come to the end of technical session 1 of this historic event. On behalf of organizer, I express my sincere thanks to Professor D.J. Bhagiraj sir, former President of Mycological Society of India, to chair the session and giving their valuable time. Thank you very much, sir. I am also thankful to Professor Naveen Kango sir for their presence as a co-chairperson for this technical session. Thank you very much, sir, for your presence. My special thanks to Professor Bernard Rodrigues, sir, President Mycological Society of India, for delivering informative and enthusiastic lectures on a very interesting topic, that is the mycorrhizal fungi. Thank you very much, sir, for your nice presentation. I thanks to Mycological Society of India and all office bearer of the MSI for their constant support during the organization of this event. Thank you, one and all. I am very much thankful to Principal Shri Shivaji Science College, Amravati, Organizing Secretary of this event, Dr. Dilip Hande sir, Joint Organizing Secretary, Professor Magirwar Madam, and all the teaching and non-teaching staff of this department for taking painful efforts for the organization of this event. Lastly, I thanks to all those who helped me for the grand success of this event. Thank you one and all. Now, I hand over this mic to the Dr. Sushita Kodke madam. Thank you one and all.
Thank you, sir. Good afternoon, all of you. Honorable dignitaries, office bearer of MSI, President, Secretary of MSI, learned scientists, respected teachers, and dear students, a very good afternoon to one and all. I, Dr. Suchita Khorke, Assistant Professor, Vinayak Vignan Mahavidyalaya, on behalf of Organizing Committee of First World Fungus Day 2021, feel privileged to welcome you all for the technical session 2. It is well said that the aim of education is knowledge, not of facts, but of values. I feel it's like a consortium of experts of mycology, and we have been listening to the enchanting and enlightening session of many experts. So let's continue the joy of getting enlightened by these eminent speakers. I welcome you all in the technical session 2. For this session, we are very fortunate to have Honorable Professor N.S. Atri, former President and former Editor-in-Chief, Kawaka, MSI, and Department of Botany, Punjabi University, Patiala, as a chairperson for this session. I request Professor N.S. Atri kindly chair the session. I also welcome Dr. Aniket Gade sir, Assistant Professor, Department of Biotechnology, Sant Gadge Baba, Amravati University, Amravati, as a co-chairperson. It gives us immense pleasure to welcome speaker for this technical session 2, respected Professor Rupam Kapoor, past president of MSI, Department of Botany, University of Delhi, Delhi. Today she is delivering her lecture on the topic Botrytis cinerea, a broad host range fungal necrotroph insight from forward genetics. So it is great honor and pleasure for me to introduce before you the chairperson of today's session, Professor Dr. N.S. Atri sir. Professor Narendra Singh Atri sir, born on 1st September 1955 in the remote village of Tehsil Pachad, district Sirmaur in Himachal Pradesh. Initially, he worked as a lecturer in botany at SMD RSD College, Pathankot from 1982 to 1987. In 1987, he joined as a lecturer in botany department, Punjabi University, Patiala, and subsequently he became a reader in 1991 and professor in 2004, the position on which he worked till August 31, 2020. During his professional career, spanning over more than 14 years, Dr. Atri sir participated in more than 60 national and international conferences, in many of which he chaired and co-chaired different scientific sessions. Dr. Atri sir is an accomplished mushroom scientist with national and international recognition. So far, he has supervised 34 students at postgraduate level for MSc, 10 students, MPhil, 10 students, and under his supervision, 14 students received PhD degree on mushroom-related research problems and completed 3 research projects funded by University of Grant Commission. At present, he has published 180 research papers in the national and international journals of repute and edited and authored 7 books along with his students and collaboration. During the past 14 years, sir, along with his students and national and international collaborators, taxonomically described over 700 mushroom taxa belonging to the different families. Out of the described taxa, 45 species and varieties were new to science, and about 25 taxa were described for the first time from India. Simultaneously, number of checklists, including checklist of Indian Russelaceae, checklist of genus Lentinus, checklist of a number of genera, and the documentary of Agromycetious mushrooms of India was also published. Dr. Atri sir has contributed significantly on the nutritional, nutraceutical, socio-biological, domestication, and conservation aspects of indigenous strains of wild edible mushrooms and some entomomorphopathogenic fungi collected from the northwest India. Some of such mushroom on which domestication studies were undertaken are Lanthanus squarosolus, Lanthanus conatus, Lanthanus torultellus, and Lanthanus cladopus, etc. In the field of ectomycorrhiza, Sir, along with his collaborator, identify as many as 22 potato mycorrhizal mushrooms associated with the associate of the Shoria robusta in Shivlik range of the Himachal Pradesh and Uttarakhand, and published the morphoanatomical details of their ECM root. Dr. Atri sir is a life member of Indian Botanical Society, Meerut, Mushroom Society of India, Solan Mushroom Growers Association, Solan, Phytopathological Society of India, New Delhi, Society of Mycology and Plant Pathology, Udaipur, Punjab Academy of Sciences, Patiala, HIM Science Congress Association, Solan, and Mycological Society of India, Chennai. He served as a councillor 2011 to 12, Vice President 2012 to 13, and President 2019 of Mycological Society of India, beside being on the editorial board of number of prestigious journals, including Mushroom Research, Journal of Mushroom Society of India, Nilambo, Kawaka, Journal of Mycological Society of India. He also served as the sectional editor for the Indian Phytopathology, first editor and then editor-in-chief for Kawaka, being transaction of Mycological Society of India, and reviewer for the Mycotaxon, Taiwania, Indian Phytopathology, Kawaka, Indian Journal of Forestry, and many other national and international journals. His achievements have been recognized by different awards, including a prestigious Agnihotridu Memorial Award and Professor K. Natarajan Memorial Award by the Mycological Society of India, Chennai, and honorary fellowship by Indian Mycological Society, Kolkata. Dr. Atri sir was honored by Goa State Biodiversity Board in recognition of the contribution made for nurturing the ideals of mushroom biodiversity conservation and sustainable utilization. Sir served on the UGC SAP Advisory Committee of Usmania University Botany Department from 2016 to 20. Advisor DPT Government of India sponsored Star College program at Deoba College, Jalandhar from 2012 to 20. Member of Research Advisory Committee RAC of ICCAR, Directorate of Mushroom Research, Chambaghat, Solan, Himachal Pradesh, 2019 to 22. Member of Committee for the Third Party Independent Evaluation of the Ecto Test program by MOEF and CC Government of India. In recognition of his significant contribution in mushroom mycology, two mushrooms, namely Lactarius atrae van D. Putti and K. Das Roselia pularis, were Atri, K. Das SL, MI Mill, and J.R. Sharma has been named after him. So today we are very fortunate, such a profound scientist among us today. Welcome, sir, for this technical session. May I now request Dr. Prashant Deshmukh, sir, to please introduce the co-chairperson of today's technical session to Dr. Aniket Gade, sir.
Thank you, madam. First of all, I am very much thankful to the organizer secretary who gave me an opportunity to introduce our co-chairperson for technical session 2, Dr. Aniket Kumar K. Gade, sir. Really, it's a matter of great honor and privilege to introduce a stalwart, genius, and hard work personality in the field of nanotechnology and microbial technology. Sir, you hardly need any introduction. Sir is currently working as an assistant professor, Department of Botany, Sant Gadge Baba Amravati University, Amravati. She has also worked, he has also worked as a junior research fellow under the able guidance of Dr. D.R. Ranade at Agarkar Research Institute at least for 1 year. He has also served as a full-time lecturer on permanent basis in bioinformatics at Vidya Pratishthan Arts, Science and Commerce College, Baramati, at least for 2 years. Currently, sir is working as a associate professor in the Department of Biotechnology, Sant Gadge Government Institute of Science. He has also worked as an associate professor at Department of Biotechnology, Government Institute of Science, Aurangabad, for 6 to 8 months. As far as academic qualification is concerned, sir has a great academic performance right from the graduations of and even in postgraduate also. He did post-graduation in biotechnology as well as graduate in microbiology, chemistry, botany from Sant Gadge Baba Amravati University, Amravati. He did PhD under the able guidance of Professor M.K. Rai, Head Department of Biotechnology, Sant Gadge Baba Amravati University, Amravati, and pursued his PhD on topic microfabrication of silver nanoparticle by using different FOMA species. He has also 1 year of postdoctorate research works at Biology Department, Utah State University, Logan. He has completed several research projects sponsored by various agencies like UGC, DST, Ministry of Environment and Forest, DRDO, and RGSTC. Overall, the sir has performed his work mostly in the area of biosynthesis of silver nanoparticle, then screening of different fusarium species for synthesis of silver nanoparticle and their potential for development of novel antimicrobial agent, then diversity of endophytic fungi in selected medicinal plants of Meerkat forest, along with extraction and evolution of potent natural product of medicinal plant of Meerkat forest for blocking viral enzyme. Overall, the project cost is comes to near to 4 crore during his entire academic work. Sir has also got studentship for advanced diploma in bioinformatics from the Department of Biotechnology, Government of India in 2001 and 2002. He has also a recipient of INSA Summer Research Fellowship in 2008 and also awarded with Young Scientist Award in 2010 in the National Conference on Microdiversity with its sustainable exploration and biotechnological application and 30th annual meeting of Mycological Society of India. He has also worked as a deputy coordinator for UGC SAP DRS 1 from 2012 to 2017. He is also a recipient of international award from Biotechnology Advances elsewhere, top cited articles for 2009 to 2011. He has a postdoctorate Raman Fellow who has visited USA for the year 2013 and 14 and also received a long-term ICMR DHR International Fellowship for Young Biomedical Scientists 2019-20. He has also visited University of Campinas, Brazil, under Indo-Brazil Collaborative Research Project work, and also Utah State University for his postdoctorate research work. Under his able guidance, one student has awarded PhD degree and 4 are recently working on various areas of research. So far, the 32 students has completed their dissertations under his able guidance. Sir has also got 2 patents till in the date and overall having 68 international research paper, 6 national research paper and 18 books in chapter. The sequence of 34 ITS sequences submitted to the NCPI and the credit is also in the name of our co-chairperson. So with this, I hand over the mic to Khorke madam. Please, madam.
Thank you, sir. To proceed further, may I now request Chairperson of the session, Honorable Professor Atri sir, to take over the proceeding of the session and introduce Professor Rupam Kapoor, madam. Thank you, sir. Over to you, sir.
Thank you very much, ma'am, for the nice introduction. Good afternoon. Namaskar to one and all in this webinar on this auspicious day. At the outset, I am extremely thankful to MSI and to Dr. Hande and the management of Shri Shivaji Science College, Amravati, for providing me this opportunity to be part of this auspicious event and for organizing a fungal awareness week culminating into World Fungus Day. In India, we have been celebrating 2nd October as Gandhi Jayanti to mark the birthday of our father of nation, Mahatma Gandhi, and also our former Prime Minister Lal Bahadur Shastri, as Professor T.N. Lakhanpal has rightly mentioned. It is, I think, for the first time in my knowledge that 2nd October is being celebrated as the World Fungus Day in India by MSI. My felicitations to you all participating in this event today. The week-long awareness program chalked out by the organizers in collaboration with MSI speaks volumes about the thought process that has gone into it. The two lectures by eminent mycologists today adds more academic flavor to the celebrations of this auspicious day. We have just listened to the excellent lecture on mycorrhiza by President MSI, Professor B.F. Rodrigues, and the second lecture in the series is by equally eminent mycologist who was immediate past president of MSI, Professor Rupam Kapoor of University of Delhi. She is a distinguished mycologist and plant pathologist who has made significant contributions in the field of arbusculus mycorrhiza, mycorrhizal technology, and molecular interactions of plants with symbionts and pathogens. So far, she has supervised 10 students for MPhil and 12 students for PhD and completed 5 R&D projects funded by different funding agencies. During her academic career, Professor Rupam Kapoor has published several research papers in journals of high impact and several awards. She has several awards to her credit. She was awarded Women Scientist Fellowship under WASH scheme of DST in 2004 to 2009. DST was cast fellowship from 2008 to 2009 on plant microbe interaction at Department of Biology, University of Alabama, USA. Y.S. Murthy Medal 2010 for Young Scientist by Indian Botanical Society. K. Natarajan Memorial Lecture Award by the Mycological Society of India. She's associated with various professional academic societies in various capacities. And with this brief introduction, I invite Dr. Kapoor to start with her lecture, and she has already been told that she has chosen to speak on Botrytis cinerea, a broad host-range fungal necrotroph, insights from forward genetics. At the same time, I welcome Dr. Aniket Gade of Amravati University as co-chair, and I request him to kindly note down the gist of the lecture so as to present at the end of the lecture. So thank you very much. And I invite Dr. Kapoor to please start with the proceedings.
Thank you, Professor Atri. I humbly accept your introduction, which was quite elaborate. Thank you very much. Good afternoon to all. My congratulations to the organizers, Shri Shivaji Science College, Amravati, and the organizing secretary and the principal of the college, Dr. Hande and Dr. Professor Chaudhary. I would also like to congratulate Professor Raman for managing simultaneously the celebrations going on at various parts of the country. He is managing it all simultaneously, while we are thankful that his presence is here in this meet. Professor Rodrigues, it is always a delight listening to you. Professor Lakhanpal and all. We all are celebrating World Fungus Day today, and celebration of fungus is celebration of mycologists. And today we are also celebrating the birth anniversary of Mahatma Gandhi, Lal Bahadur Shastri, but one more eminent mycologist whose birthday was yesterday. I want all of you to please congratulate him and wish him. It was Professor Lakhanpal's birthday yesterday. We wish him a very long and healthy life, and we pray to God that you continue to guide us through to the right path. With this, I start my presentation. My presentation today is total contrast to what Professor Rodrigues has spoken about. It is about a fungus which is totally starkingly different from mycorrhizae, the fungi that form symbiotic association with 90% of the terrestrial plants and have friendship with them for last 400 million years ago, and have been extending their benefits to the host plants in various ways and indirectly serving the mankind. The fungus which I have chosen today, it is a broad host range necrotroph. It is a ruthless brute which has no regard for these plant species, and it has got no regard for the stage at which the plant is. It just infects it ruthlessly, and just opposite to fungus, which is very friendly, and it almost gets into friendship with most of the plant species. With this, I go ahead that pathogens are categorized on the basis of their nutrition as biotrophs, necrotrophs, and hemibiotrophs. Biotrophs are the ones which colonize the living cell and derive food and energy from them, whereas necrotrophs are the ones which kill the host cell and then derive food and energy from them. And intermediate among them are those hemibiotrophs, to which Magnaportheorizae belongs, which initial phases it survives in the living cell, whereas in later stages it gets converted into a necrotroph and then derives food from dead cells. So today's fungus that I have chosen is Botrytis cinerea, which has a very broad host range, and it is a necrotroph pathogen. Botrytis cinerea is also known as gray mold fungus. It is the second most phytopathogen after Magnaportheorizae, which is capable of infecting more than 1,000 plant species, and it occurs an annual loss of about 100 billion worldwide. High humidity, senescing plant tissue, and high sugar content, along with low temperature, are favorable for its growth. This fungus has no regard to the plant growth. It is capable of infecting a seedling to a mature standing crop, and even the products which is stored in cold storage. The symptoms of this fungus varies depending upon the host plant and the stage at which it is infecting. It starts with the water-soaked necrotic lesions, which later on form a lawn of gray mold, conidia, and necrotic spots, as can be seen on apple, or blemishes on rose and marigold. When it colonizes a plant tissue, the senescent tissue within it, there is formation of sclerotia, a melanized thick structure, which helps in perennation to the fungus to overcome the adverse conditions. And on advent of favorable conditions, these sclerotia, they germinate to form conidiophores. And when the conidia are dispensed from it and land on the surface of the leaf, it germinates and colonizes. The fungus releases a battery of toxins and cell wall degrading enzymes, hence completing the disease cycle. Normally, Botrytis cinerea reproduces asexually. Very rarely we can come across sexual reproduction. Very recently, it has been found that when Botrytis cinerea colonizes a plant tissue, plant initially remains asymptomatic. So probably the fungus is in its quiescent stage. However, on observing the tissue, it is found that it is not in its quiescent stage. Actually, it is a cryptic biotrophic phase during which it invades and colonizes the plant tissue, and later on then gets transformed into its necrotrophic phase, where it releases cell wall degrading enzymes and toxins so as to derive food from the lysing cells. Because of its economic importance, Botrytis cinerea has been extensively studied, and about hundreds of virulence factors that contribute to its pathogenesis are known. And most important among them is the fungus produces a lot of cell wall degrading enzymes and oxalic acid. However, because of its high evolutionary potential and its genetic flexibility, it very soon overcomes any fungicide that is applied to it. So there is need to find new target sites or new virulence factors so that new strategies to control this fungus can be developed. So whenever a plant is challenged by any stress, biotic or abiotic stress, it results in production of reactive oxygen species. An outcome of reactive oxygen species is highly dependent upon the life cycle of the pathogen. And reactive oxygen species production is also associated with hypersensitive response or programmed cell death. When there is high levels of ROS, it results in impeding the spread of biotrophic pathogen, because dead cells also kills the biotrophs which is colonizing it, whereas high levels of ROS, it further aids the spread of necrotrophic pathogen. So Botrytis cinerea produces a toxin, oxalic acid, which is a broad range toxin, and it is found to have dual role in the two phases of Botrytis cinerea. Firstly, once it colonizes, it releases oxalic acid, and oxalic acid here dampens the production of reactive oxygen species, and by changing the redox potential within the host, as a result, subduing the host defense response and also autophagy. Once the fungus transforms from its biotrophic phase to necrotrophic phase, now this time oxalic acid functions entirely differently. It overwhelms the production of reactive oxygen species, at the same time decreases the pH, which helps or felicitate the cell wall degrading enzymes and also chelates calcium, hence resulting in dead apoptosis or death of the cells around it. The fungus, when it colonizes, it releases a lot of cell wall degrading enzymes, but pectinases are the hallmark enzymes of Botrytis cinerea. Botrytis cinerea releases several pectinases such as pectin lyases, endo-exopolygalactonurases, pectin methylesterases, and it has been found that endopolygalactonorase is required for full virulence of Botrytis cinerea. At the same time, what oxalic acid does, it chelates the calcium ions that are acting as cross links between the polygalactonorange chains of pectin. So once the oxalic acid, it chelates the calcium. As a result, there is gap created between the chains of polygalactonorans. As a result, polygalactonorases, the enzyme, it enables or it facilitates the action of PG. Moreover, the activity of PG, polygalactonorases, is much higher when the pH is low. Botrytis cinerea, it is a model pathogen. Firstly, like I said, it's the second most important phytopathogen based on its scientific and economic significance. And also its genome has been sequenced. Its genome is of very small size. It is haploid in nature and reduced repeat content is very less. So as a result, it is highly amenable to any genetic or molecular manipulations. We isolated a virulent strain of Botrytis cinerea and we tested its virulence. This strain was highly virulent and it had the ability to form sclerotia. What we did to gain random mutants, we did random mutagenesis, insertional mutagenesis using Agrobacterium cumefaciens mediated transformation. As a result of that, we got 800 insertional mutants. On first round of screening for pathogenicity, we got 200 mutants that were impaired in virulence. And after several rounds of screening, we narrowed our search to 4 mutant strains that were highly impaired in virulence. The pathogenicity assay was done in a National Phytotron Facility, IRI, and it was found that out of various mutants that were screened, BCM55 showed 94.5% decrease in virulence, whereas BCM27 showed 92% decrease in virulence. BCM29 showed 79.4% decrease in virulence compared to wild type, whereas mutant strains BCM 425 and 780 were completely avirulent. They were not able to cause disease at all. The transformants were confirmed by PCR, by fluorescence microscopy, and number of copy inserts were ascertained by southern blot hybridization. And the tagged tDNA flanking region, that tDNA goes and inserts into a gene and stops its expression. So the flanking regions of tDNA are ascertained by thermal asymmetric interlaced PCR, and by thermal asymmetric interlaced PCR, the tertiary gel yielded a single band which was extracted, cloned in PGMT-EZ vector and sequenced commercially. So remember BCM 425, that was totally avirulent. The gene that was disrupted by tDNA was found to be superoxide dismutase gene. This gene, this superoxide dismutase is involved in these dismutation of superoxide radical to form hydrogen peroxide, which is superoxide radical is highly lethal for the survival of any living cell. However, perusal of literature revealed that we have stumbled upon a gene whose role in virulence has already been reported earlier in 2004 and 2017, where superoxide dismutase was found to play an important role in virulence of Botrytis cinerea on French bean as well as on Arabidopsis and tomato plants. So the other mutants that genes that we found were disrupted were gene encoding diacylglycerol orthoacyl transferase 2 and nucleoporin complex protein, kinesin, and a predicted protein that was not found in whose homology was not found in any database except for Botrytis cinerea. So we carried our work on these 4 mutants, and in today's presentation, I will be limiting my discussion on only 3 mutants. First was to characterize the mutant. So for characterization of any gene for virulence, we have to again see that this gene fulfills molecular Koch postulates, so that this gene is disrupted, target deletion was carried out, it was cloned, and then this gene was reintroduced in the targeted deletion mutant to see whether its reintroduction in the deletion mutant result in rescue of A virulence in the mutant strain. These mutants were also morphologically characterized and biochemically characterized to ascertain to functionally validate this gene. Diacylglycerol is a key enzyme, which is a membrane-bound enzyme that catalyzes the conversion of diacylglycerol by transferring acyl group to form triacylglycerols that are repository for synthesis of lipids, including membrane lipids. Targeted deletion mutants were raised, as well as complemented strains were also raised, and their validation was done by determining the transcript levels of this gene in insertional mutant, deletion mutant, and the complemented clone. While there was negligible expression of this gene in the insertional and deletion mutant, the levels were stored, restored, equivalent to the wild type in the complemented clones. We determined the lipid content in these mutant strains. While there was about 29% decrease in lipid concentration, there was about almost 45% decrease in the concentration of triacylglycerols, which was further validated by TLC, where we've got highly prominent spots in wild type as well as complemented clones, whereas the spots in insertional and deletion strains were very mild or very low. Phenotypic characterization showed that there was change in the morphology in compared to moldy, cottony, aerial growth in wild type and complemented clones. There it was matty and the margins were raised in the mutants where this gene was not being expressed. But most interestingly, we found that the disruption of DGAT2 gene resulted in inability of the strains to form sclerotia, whereas reintroduction of the gene in the complemented clone again reinstated the ability of the fungus to form sclerotia. Like I mentioned, sclerotia play an important role in survival of hostile environment. They act as primary inoculum in disease cycle, and they play an important role in sexual reproduction as female parental tissue, which is fertilized by microconidia from opposite mating type. Phenotypic characterization showed there was no difference in conidial size, whereas there was 40% decrease in the sporulation rate. In vivo pathogenicity assay again showed that there was less disease on these when chickpea plant was challenged with insertional and deletion mutant, whereas when they were challenged with complemented clone, the disease severity was comparable with that of wild type. In vitro virulence assay further corroborated the results that we observed in vitro assay. Then we also ascertained the response of mutants to various stresses, where medium was supplemented with sodium chloride or sorbitol for osmotic stress, hydrogen peroxide for oxidative stress, SDS for membrane, and Congo red for cell wall stress. And it was found that while these mutants were similar in response to the osmotic stress, there was marked decrease in growth when these mutants were subjected to membrane stress or cell wall stress. Plant response to these mutants was also ascertained in terms of reduction in concentration of chlorophyll, oxidative damage, activity of antioxidant enzymes, relative expression of defense genes. For this, 4-week tomato plants were treated with conidial suspension of wild type, and mutant conidial suspension controls were sterile water. Fungal load was determined by determining the relative abundance of cutinase A gene. While there was continuous increase in fungal load in the wild type, there was no increase in the proliferation of fungus within the host cell up to 72 hours post-inoculation in wild type in which the gene was not being expressed. These observations were also corroborated by disease severity. While there was steady increase in disease severity, the progression of disease in the mutant, it was very slow. The production of oxalic acid was decreased by 50%, and correspondingly, the polyglucoronase activity also decreased 25%, while there was not much change in pectin methylesterase activity. The defense response elicited by the host plants was much higher in wild type compared to the mutant. With respect to all, I will not be going into detail considering I have to catch up with the time. There was a decrease in the enzymes that were responsive to salicylic acid, such as PR1 chitinase, which belongs to PR3 kind of protein, and hypersensitive response gene, whereas the expression of jasmonic acid responsive gene, proteinase inhibitor 2, its expression increased in wild type, in a mutant type compared to wild type. What could be the reason for this? We've yet to find out. So what all results we got, we can summarize. We have proposed a model where in the wild type, when DGAT2 is being expressed, diacylglycerol is converted into triacylglycerol. So this pool of triacylglycerols are used that form robust hyphal growth, intact fungal membrane, whereas in mutant in which DGAT2 was not being expressed, there was decrease in the pool of triacylglycerols. As a result, there was aberrant hyphal growth, deformed fungal membrane, reduced mat-like growth, no sclerotia formation, impaired penetration, incompetency to tolerate membrane and cell wall stress, and moreover, there was decrease in production of virulence factors, oxalic acid, and polyglucoronase activity that resulted in decrease in virulence in the mutant where DGAT2 was not being expressed. Coming on to the second protein, nucleoprotein complex. This protein, it is involved in selective trafficking of proteins, nucleic acid, and other factors between nucleoplasm and cytoplasm. As a result of trafficking of proteins from cytoplasm to nucleoplasm or from nucleoplasm to cytoplasm, this protein plays an important role in regulation of physiological processes within the pathogen, kinetochore and spindle assembly, DNA repair, and regulation of gene expression. Targeted deleted mutant was raised, and the expression of this gene was ascertained, which further validated that we have successful in raising deletion mutant. Phenotypic characterization was done. There was not much difference between the wild type and mutant strains. They retained the ability to form sclerotia as well. In vivo pathogenicity assay, there was no effect when chickpea plants were challenged with the mutant or as well as with the deletion mutant, whereas the disease could be seen when chickpea plants were treated with wild type. Not much discernible difference was observed when these mutants were tested for their response against stress, though it is evident in the photograph here, but statistical analysis showed a non-significant differences between the mutant strains and the wild type. There was decrease in the oxalic acid concentration. Correspondingly, there was decrease in polyglucoronase activity, and also there was decrease in pectin methylesterase activity in those strains where this gene was not being expressed. The response was also same as BCM55, so I'm skipping those slides. And coming on to the model which we propose, by which NUC 184 contributes to the virulence of Botrytis cinerea. And in wild type, NUC 184, which is a component of nucleopore complex, it facilitates the transport of cargo with help of important protein inside the nucleoplasm, and also export of certain proteins or nucleic acids from nucleoplasm with help of exportin. It helps in the transport of cargo inside the cytoplasm. This was in case of mutant wild type, when there is transport. So there was exchange of signals, information, proteins, nucleic acid. Whereas in mutant, when there is no NUC 184 being expressed, there is no transport. And we propose that probably this protein is specifically involved in some transport of some factor across nucleoplasm and cytoplasm that is playing an important role in expression of virulence in Botrytis cinerea. What is exactly being transported is yet to be elucidated. Now I come on to my third protein, kinesin protein, KLP7. Kinesins are microtubule-based energy hydrolyzing motor proteins that facilitate cell division and intracellular transportation. So KLP7 kinesins are involved in intracellular transportation, and KLP7 is a unique kinesin in the family kinesins that is found only in phylum Mycotina, to which Botrytis cinerea belongs. Again, we carried out targeted deletion and complementation of KLP7 encoding gene, which was again confirmed by the expression of this gene. In vitro, a virulence assay was done on detached tomato plant leaves, and it was found that there was decrease in lesion size, there was decrease in disease severity, there was decrease in conidial germination. In the deleted mutant, in the insertional mutant and the deleted mutant, whereas the pathogenicity avirulence was rescued when this gene was reinstated in the complemented clone. We interestingly found there was decrease in conidial size. There was also difference in the hyphal morphology. While the hypha was uniform and broad in wild type, it was irregular, constructed in those mutant strains where this KLP7 encoding gene was not expressing. There was also difference that the color, change in the color, the colonies of mutants were melanized. Their ability to colonize host tissue was also compromised. There was also decrease in % germination of conidia, and the germination of conidia was also delayed with respect to wild type. In these mutants also, there was decrease in oxalic acid concentration, there was decrease in polyglucoronase activity, as well as there was decrease in pectin methyl esterase activity. So we again proposed a model by which we think that KLP7 contributes to the virulence of Botrytis cinerea. In wild type, KLP7 in association with microtubule helps in intracellular movement of these secretory vesicles. And probably KLP7 is implicated in the intracellular movement of these secretory vesicles towards the apical region of the hyphae. When KLP7 is not being expressed in the mutant, there is no transfer of these secretory vesicles. Probably these secretory vesicles are carrying the virulence factors. And as a result of this, the pathogen which is not expressing this protein does not express pathogen virulence, and it loses its ability to cause disease. So with our forward genetics, we were able to identify 3 novel genes and functionally validate their role in virulence of Botrytis cinerea. This information can now be extrapolated to investigate the roles of these genes in other pathogenic fungi. With this, I thank you all for patient hearing. I would like to acknowledge the efforts of my students and thank you. May we all enjoy the benefits of fungus, but keep the devil in check. Thank you.
Thank you very much, Dr. Kapoor, for nice exposition of the information you have generated through your researches on Botrytis cinerea as a model pathogen to study the molecular mechanisms underlying the host pathogen interactions. They've identified 3 genes and they've worked out their characterization and proposed models explaining the factors responsible for virulence of the this notorious pathogen. Very elaborate lecture, and I think the audience must have really enjoyed it. And I request the audience to put up 1 or 2 queries if they have to Dr. Kapoor, or they can also discuss these with her subsequently who are interested. But for the time being, with the time constraint we have, I request the audience to put up 1 or 2 queries to Professor Kapoor. I'm sorry, was I audible?
Yes, sir, you were. It's lunch time now. We are challenging them.
Okay, okay. It seems I was muted. Could you hear me, whatever I spoke?
Sir. Yes, sir.
Yes, sir. We could
Okay,
hear you.
okay. So please, 1 or 2 queries.
I think they can contact Professor Rupam Kapoor directly. I think so.
Yeah, she explained everything.
Everything.
Experimental evidences, pathogen, in vivo pathogenicity assays, etc. Everything was self-explanatory. Otherwise,
some question is there, ma'am. Is there any future scope for fungus supplements?
Oh, yes, they are the need of the hour. There are so many experts of fungi sitting here. I am not the right person to answer when Professor Attari is chairing it. We know that mushrooms can act as a great supplement, and the consumption and the acceptability of mushrooms in India is very less. It is less than 50 grams compared to 2 to 3 kgs in countries like USA and Europe. So and various other mushrooms, various other fungi as single cell proteins, they can contribute as supplements, certainly.
Yeah, that question was from Bhargavi Rami Shetty.
they are being exploited now extensively as functional foods. All these mushrooms, because of their bioactive potential, you have a lot of bioactive components there which are of utility. Both nutritional and nutraceutical advantages are there, and there are a number of fungi which are in use and which are being exploited in this regard. So thank you very much. I'll request Dr. Aniket Gade to add
Okay. Thank you, sir.
Aniket Gade, please.
Yes. Thank you, sir. Thank you, Professor Attari, the chairman for the session. What we could listen during this talk given by Professor Rupam Kapoor, a past president of MSI, Department of Botany, University of Delhi. It was a nice and elaborative introducing about one of a notorious fungus Botrytis cinerea. Now, what could be a better platform than the platform of World Fungus Day to give us an elaborative way? What was interesting about a talk was ma'am could, in a simple language, could explain how we can characterize a mutant and go into the in-depth study identifying the gene level which will be responsible with respect to the virulence of that particular fungus. So all the sophisticated techniques which were used in the study were put forth by ma'am in a simple language. I think that is a key thing which we can take out from this lecture. Ma'am has explained with respect to the three genes they have characterized and also proposed the model how they could help with respect to host pathogen interaction. So what we teach in a class about the role of mutants and the mutagenesis has helped us to identify different aspect about gene. The same features were applied by ma'am, and in her studies, it's really, it could be reflected in a very well how she could propose even the models in a better way. So probably this is what it was a lecture which was enjoyed thoroughly by the student, how a complicated things can be put up in a most lucid language. So I would like to thank ma'am for this particular talk, even about the technique she has helped. And apart from that, even I would like to thank the organizers because this is World Fungus Day, which has been celebrated. And we all are here to encourage the students to get into and trying to understand more aspect. There are many hidden things about the fungus which are not been known. And today, Madam has helped us to give a focus on one of such fungus like Botrytis cinerea. So thank you, ma'am. Thank you, organizers. Thank you for such talk. Thank you.
Thank you, Dr. Gade, and
Thank
thank
you.
you, Professor Ratri. And I take cognizance of Professor Yashpal's presence here. Thank you,
Yeah.
sir, for joining us for some
Yeah,
time.
Professor Yashpal has joined. Welcome, Professor Yashpal. On behalf of MSI, I thank Professor Rupam Kapoor and Professor Ratri and Dr.
Aniket.
Yeah, from Amravati University. We for been co-chairing the session. Hande, over to Hande.
Aniket.
Okay, okay.
Yeah.
Thank you, sir, for your remark and kind words. Before we conclude, may I request Dr. Prashant Deshmukh, sir, to propose the vote of thanks for this session.
Thank you, madam. We are very much honored by gracious presence and word of guidance and brief lecture by Rupam Kapoor. Also, we are very much honored for chairperson Professor N.S. Atri, former president and former editor-in-chief, Kavka, MSI, and Department of Botany, Punjabi University, Patiala, Dr. Aniket Gade, Associate Professor, Department of Biotechnology, Sant Gadge Baba, Amravati University, Amravati. I assure your valuable word and lecture by Professor Rupam Kapoor surely ignites the mind of billions and encourage to work in the area of Kavka for the benefits of living society as well as for mankind. I once again thanks on behalf of whole team of Department of Botany, Shri Shivaji Science College, Amravati, and hope for your future cooperation. Okay, so now it's a most awaited moments for we all. It's a time for valedictory program and winner declaration. So for this, now I invite uh, Professor T.B. Wankhede sir and as a co-host Dr. S.V. Punkar madam. Please, sir.
Good afternoon, everybody. Good afternoon and greetings of the day. Firstly, I pay my homage to the conscience of education, Santa Gadge Baba, and also the founder of Shri Shivaji Education Society, Bhosai Punjabrao Deshmukh. Friends, on the behalf of Department of Botany, Shri Shivaji Science College, Amravati, I, Professor Tushar Wankhede, and my colleagues, Dr. Swati Punkar, welcome all office bearers of the Mycological Society of India, all the collaborating partners of different colleges, guests, faculty members, and most important students for this valedictory function on the World Fungus Day. This World Fungus Day celebration is a joint venture of Sri Shivaji Science College, Amravati, and 12 collaborative college under Sant Gadge Baba Amravati University, Amravati, and one college is out of the state. Friends, firstly, I would like to welcome the president of today's function, Professor P.W. Devtare sir. Dr. Devtare sir is a former head of the department of our botany department. So, sir, welcome. It's a matter of immense pleasure to welcome chief guest of today's valedictory function, Professor Munruchi Kaur, head department of botany, Punjabi University, Patiala. She is also vice president of Mycological Society of India. It's my privilege all members of the organizing committee and my colleagues, Professor G.B. Hedau, Professor R.C. Magirwar, Professor D.D. Khedkar, Professor A.N. Darsimbe, and Professor P.D. Deshmukh. Welcome all. Friends, why to remember fungus? And second, why we required a recognition to the fungi. Whitaker, 1969, put the perception of the separate world of the fungal kingdom. We cannot imagine the life without this tiny wonder of the nature that is the fungus. We in the 21st century witness fungal research with the incredible speeds. Since a long time of progression, we are the spectator of fungal world in the terms of staple food, in the terms of marvels of medicine like the penicillin as a wonder drug, and also nurturing ecological schemes like when algal association took place with the fungi and formed the lichens. And even more perspectives, the gene silencing mechanism in Neurospora crassa at a molecular level. So, friends, not only this, but fungus can withstand as a parallel silent partner of symbiosis like mycorrhizals and also in the field of agriculture. In recent, the whole world catch consciousness about the many pathogenic fungi like mucormycosis during the COVID-19 pandemic. So friends, although some opinions about the fungus being colored by the menacing undertones from the stories and knowledge that a fungus can rot, fungus can damage, fungus can kill or it may be a poisonous, but our planet's ecosystem would not be function without the fungi. So there will be no fertile soil, there will be no plants, no herbivores, no carnivores, no humans can survive without the fungi. Okay, friends, this program is live online on our college website and also on the platform of Zoom app and along with the YouTube streaming. So kindly stay connected with us. So without taking your time, friends, this event is celebrated by the enthusiastic collaborators who made historic contribution to the recognition of the World Fungus Day
To know about this event, I would like to invite Professor D.V. Hon'ble, organizing secretary of this event, to figure out the summary of events in a comprehensive sense of compilation. Professor D.V. Hon'ble.
Good afternoon, one and all present here. Dignitaries on the dais, President of today's chairman, our ex-head of the department, Professor P.W. Devtare, our head, Professor B.K. Dorkar, my colleagues, Dr. Ganesh Hedau, Dr. Rekha Makirwar, Professor Tushar Wankhede, Dr. Prashant Deshmukh, and all from the Shri Shivaji Science College. And those my senior mycologists which are present in the online platform, President of Mycological Society of India, Dr. Rodrigues, Secretary, Professor Raman, Professor Atri, Professor Lakhanpal, Professor Bhagyaraj, Professor DJ Bhatt, Professor Janardhanan, Chief Guest of Valedictory Function, Professor Munruchi Kaur, Dr. Aryan Karwar, Professor Ishpal Sharma, and all present here. It's a matter of immense pleasure to share with you the experiences since our college decided to organize the celebration of World Fungus Day 2021 and Fungal Awareness Week in association with Mycological Society of India. First, I would like to thank Mycological Society of India for accepting our proposal for the organization of this event. As soon as this opportunity knocked our door, we tried to communicate the announcements like with the participants through all online modes and the tools like Telegram, WhatsApp, email, Facebook, and all the media we have used. We have received the remarkable response for this workshop, for this event, which has boosted our morale and encouraged us to organize similar event in future also. The online event was open for all, which lot of senior mycologists, professors, UG, PG, and PhD students from India, which are registered and participated in this event. I feel proud to tell you that more than 1,000 participants registered for this celebration. Let me brief about this event. We received the outstanding responses from 12 colleges who have contributed a lot for the organization of the activities, like photography competition organized by Indira Mahavidyalaya Kalam on the world of fungi. The total participants participated to 46. Essay competition on fungi for industry organized by Sri Shivaji Art Commerce Science College, Mutara, and the participants were 129. Drawing competition on the topic Draw Your Favorite Fungus, organized by Vinayak Vignan Mahavidyalaya, Nandgaon, Kandeshwar, Amravati. Total participants participated 329. E-poster competitions on fungi, Boon to Mankind, organized by Kapoor Butle College, Digras, Yavatmal, and the total participants participated to 103. Quiz competitions on fungal systematics and applications organized by J.D. Patil Sangrudkar Mahavidyalaya, Daryapur, and the total participants were 559. The photography competitions on fungal disease on fruit and vegetables organized by Rajeshri Shahu Science College, Chandur Railway, and the total participants participated 75. The mushroom in kitchen recipe and preparation competition organized by Brijlal Biani Science College, Amravati, and the total participants were 34. Online seminar competition on fungal pathogen of human, animals, and plants organized by Sri Shivaji College of Art, Commerce, and Science, Akula. The total participants were 51. Online quiz competition organized by Government Art and Commerce College, Raipur, Chhattisgarh. The total participants were 649. online mycologist biography competition organized by SPM Gilani and Art Commerce College, Ghatanji Dist, Yavatmal, and the total participants were 70. Online fungal Rangoli competition organized by Sri Shivaji Art Science College, Chikli, and the total participants were 48. And the last online workshop on trichoderma, a biological control, organized by Sri Shivaji College of Agriculture Biotechnology, Amravati, and the total participants were 110. So I would like to tell you that the total 2,369 students directly participated in the different event organized by different colleges of this region. So today, 2nd October, after the inauguration of the online event at the hands of Professor Honorable T.N. Lakhanpal in the chairmanship of Honorable Principal G.N. Chaudhary, Principal Shivaji Science College, Amravati, and Professor B.F. Rodrigues, President, MSI, and Professor N. Raman, Secretary, MSI, and the convener of all the event. The first technical session was organized and it was chaired by Honorable D.J. Bhagiraj and the speaker, Dr. B.F. Rodrigues, President, Mycological Society of India. The second session, Honorable Professor Rupam Kapoor delivered a talk on the topic Botrytis cinerea, broad host range fungal necrotroph, insight from forward genetics. I am highly grateful to all the speakers for their valuable guidance, all the chairmen of the for sparing their valuable time to chair the session and their noteworthy remarks. The response of the student to this event indicated that the youth have innovative and creative idea, and this event provided them the required platform. In the form of organizing awareness program, we often provide them the unique chance to exhibit their ideas. The student interest in mycology are excellent. They will achieve great success in the field in future. On behalf of Sri Shivaji Science College and Mycological Society of India, I am thankful to everyone. We have tried our level best for the grand success of the event and hope we have succeeded in it. I assure you that we all would be prepared with zeal and enthusiasm to celebrate the next World Fungus Day 2020. Thank you all once again. I wish you all the success ahead. Thank you very much. Thank you.
Thank you, Dilip. Really, we all are overwhelmed to see the huge responses has been received during the celebration of this World Fungus Day. And in the next year, not 20, it is 2022. We will again like to celebrate this event under the aegis of the Mycological Society of India. Now I would like to invite Dr. Swati Punkar for the next program.
Thank you, sir. Now the time for the feedback from the participants. The various activity get huge response from the scientific and academic fraternity. Definitely, this valedictory function will not be complete. We will not get the views from the participants. To know about the views, let's invite the few participants for their delightful feedback and experience about the World Fungus Day celebration. So, I would like to call Dr. Ahmed Abdul Halim Khan, Chairman, BOS, Botany, Telangana. Dr. Ahmed Abdul Halim Khan.
Good afternoon. Good afternoon, everybody. From north to south, east to west. So the eminent mycologist of the entire country, entire nation has come together to witness a very beautiful day in the history of Mycological Society and the Shivaji College in collaboration with several other institutes for arranging such a wonderful program on World Fungus Day. I think just because of the online programs nowadays going on, we could come to know several new things that such kind of day is also marked World Fungus Day. So when I saw the flyer of this program, the flyer was of 7 pages. I was very much happy to see that the World Fungus Day is celebrated on such a large scale, and Mycological Society of India has come forward, and it has supported the group of institutions to mark today. And all the events what has been conducted are very important to make many more milestones among the young generation, bring the role of fungus in the day-to-day life, and what are the progress going on in the particularly field of mycology. So it was a very good experience of today when I could attend all the proceedings of today's online meeting. Though it is both version online, offline is going on. So I express my deep sense of gratitude for all the organizers and eminent mycologists of India for arranging such a wonderful meeting. And we were already well known of Professor Atri because we have invited for one of the talk from Telangana University supported one meeting, environment based. He was there with us. So, and many more people. Last year when Punjab University has conducted the annual symposia, at the time also I could see many people from Punjab University. So overall the day of World Fungus Day has come with good lectures of 2 eminent personalities today. So I congratulate the society as well as the group of institutions for holding such a good informative meeting online. And I just was seeing that there is even October 15th marked as Mushroom Day. So hope maybe again one more meeting may be there in this month. I think hope Mycological Society will organize Mushroom Day, also National Mushroom Day, International Mushroom Day is also coming up. So thank you for giving me this opportunity. I express my deep sense of gratitude towards Hande sir, who was very kind enough to give me this opportunity to speak in today's meeting. Thank you so much.
Thank you, Dr. Ahmed sir, for your valuable feedback. Now I call upon the Nadira Ashraf, Department of Botany, University of Kashmir.
Good afternoon, everyone. It has been a matter of great privilege to be a part of this wonderful event. I'm highly grateful to the whole team of organizers and speakers for their wonderful talk on various topics. I'm looking forward for more such wonderful events in the future, and I have myself actually participated in this event and in the past events, and I'm looking forward for more such events in the future. Thank you very much.
Thank you, Nadira madam. For the next session, I hand over the mic to Vankar sir.
Thank you very much. Ladies and gentlemen, it's a time for the presidential remark. So today we have with us our ex-head of the department, Professor P.W. Devtare sir, who made a very significant contribution in the development of this department and the field of mycology. The main mycorrhizal was introduced by Dr. P.W. Devtare in our college as well as in the university. And most remarkable event was conducted in 2012 when the 38th annual MSI meet was conducted and hosted by Shri Shivaji Science College, Amravati. And this is also initiated by the grace of Honorable Professor Sri Manurachari sir. So I would like to invite Professor Devtare sir to express his views on this event.
Good afternoon to one and all. And thank you, Tushar. First of all, I pay my homage to the great national leader of the India, Rashtrapita Mahatma Gandhi, and the Lal Bahadur Shastri. At the same time, the founder of this education society, Dr. Panjabra Deshmukh, and at the same time, the founder of the Mycological Society of India, the director, Dr. Subramaniam. So, to this function, the convener, Professor B.K. Dorkar, the organizing secretary, Dr. D.V. Hande, MSI president, Dr. Rodrigues, the secretary of Mycological Society of India, Professor Raman, and all the dignitaries, those who are joining this online World Fungus Day, Professor Yashpal Sharma, Dr. Bhagiraj, Dr. C. Manorachari, Professor Lakhanpal, Dr. Atri, and all renowned scientists, those who are present today, and all the participants and the delegates, those who joined this, and all the students, and even some of the peoples from the society also joined this particular program. So mentioning all these people, first of all, I congratulate the winners and the participants. The winners, those who have participated and bagged the prizes, and the participant for that this is the first stage of all the students, those who have participated into the different events. So participation in any particular competition is the first stage, the first attempt to go towards the winning side. So the participation is very important. Unless you participate, you cannot win. So I must congratulate all the participants, those who have participated in this particular program. Now, one thing is that this COVID-19, I take it positively. And this COVID-19 has given us an opportunity to come together, though we are online. So the most of the participants from the every corner of the nation, they can join without, and this is important, that without any expenditure, without any money. I know that when we organized the one conference in 2012, the Mycological conference, with the collaboration of the MSI, I remember that we have collected near about 5 to 6 lakhs rupees for the organization of the conference. Now you just remember, and today, with a less effort and with a less money, finance, we can conducting this national level event. So, thanks to the COVID-19. Now, here the main aim of this Fungus Day is to spread of the knowledge of the fungi in the welfare of the society and the common people. So for that purpose, I congratulate the initiation taken by the British Mycological Society. First of all, I congratulate them. And secondly, I congratulate the Mycological Society of India. They had taken interest to collaborate with the British Mycological Society and conduct this particular Fungus Day in India. And this is the very historical event that the Mycological Society of India had given an opportunity to the Shri Shivaji Science College, Amravati, and particularly to the Department of Botany. And this responsibility has been shouldered by Dr. Hande, So I congratulate Dr. Dilip Hande, who is a very enthusiastic person, is a very hardworking, very sincere. He had shouldered this particular event, and during the comments given by the Dr. Hande, he said rightly that number of colleges are collaborated with this particular event. Number of students are collaborated with this event. And the peoples from the society, they come to their colleges to see their exhibitions, to see the paintings, to see the rangolis, to take the advantage of the test of the mushroom recipes. So this, I think that more than enough that we are in a state toward to popularizing the fungi. So here, as per the Gandhiji pointed out that we have everything on this earth, but you should have to explore it. That is the main thing. So here, that and even I remember that when there is a talk in between the our Prime Minister Narendra Modi and the President of America, Joe Biden, when they are talking, that was the bilateral talk, and when they was talking, our Prime Minister said that we are the trustee of the world. What does it mean by trustee? Trusteeship is the concept which has been formulated by our Rashtrapita, Mahatma Gandhi. And the trusteeship is that concept that whatever you have in excess, that is whether it may be the money, whether it may be the land, it should be given to the persons, those who are actually needy. So I think that we are taking that trusteeship. We must follow that trusteeship because we are the knowledgeable people. We ourselves can say that we are the knowledgeable people. Then why should not we spread this knowledge? Why should not we distribute the knowledge to the society in the form of the different programs like the exhibitions in your area or the neighboring areas, and well acquaint the peoples by this particular knowledge. And I know that the facts are these. And the facts are these, that even in our home, the peoples are there. In the rural area, the peoples are there. Do they know about the fungi? No, actually they don't. What we inhale is the spores of fungi. What we eat are the spores of fungi. What we drink are the spores of fungi. But whether they are the detrimental spores, the harmful spores, or the beneficial, we don't know. Because simple example is that when anybody bring the bread, and all we know that the fungus grows over there. But if that fungus is the toxic fungus, and if that rural people or the common people, even though the persons who are the literate persons, they don't know about this fungi, they just shared the bread and spread the millions of spores into the atmosphere. Maybe the toxic one. So they are distributing the poison in the society. They don't know about these fungi harmful effect or the food poisoning. They shed the spores and they eat that bread. So this is the condition. This is the ground level situation. So being a scientist, we are the trustee. So pass on this knowledge, not just for the earning. I will say that we always see that our bank account regularly. We see the bank account regularly, that how it is increasing. But can we see that our knowledge account is growing up? So I humbly request all the persons or the students, those who are coming into this particular profession, that one should be very careful about our knowledge account. Band account, automatically it will, after a month, it will come to you. There is no any doubt about that one. And after that, I will say that our college has a tradition. Our college, especially Shri Shivaji Science College, of the very renowned the Shri Shivaji Education Society, which has been founded by the late Dr. Punjabrao Deshmukh, the agriculture minister of the India, has a vision. What he having the vision? That my children should learn here. My children get the knowledge from here. So for that purpose, he established the large number of the institutions, and the number of institutions is the largest, second largest in the state of Maharashtra. You'll find that near about 236 institutions are here, which has been founded by late Dr. Punjabrao Deshmukh. And the main intention was that you should extend your knowledge to the society. And that was the principle of the University Grant Commission, that there should be the teaching, there should be a research, and there should be an extension. And very important is that the extension is there. If no extension is there, the knowledge will be the stagnant one. And you know that the order of the stagnant water. So keep it fresh. All the times the knowledge should be very fresh. And our college work in that particular direction. Large number of the extension activities are conducted since 1977. Though this was an undergraduate college, even though I must mention the name of our the former principal, Dr. V.R. Gurude. All the principals were great. But Dr. Gurude, he has established the research laboratory here in the 1977. And see that even though he was the principal and he was engaged in the academic activities, still he was having one microscope in his cabin. And number of times I have seen that he was observing the fungi. He was observing the fungi. And I number of times I asked, sir, you get the time to concentrate yourself to see the minute characters of the spores there? He just the laugh and say that, yes, something I am doing. So here, that particular specializations of the mycology started from the 1977, and the research laboratory was established here by the Nagpur University then. And again, one thing I should tell you, that the first time in the history of state of Maharashtra, that research laboratory was having the research post of the research assistant. And it was the first post. And after that, the government has not sanctioned any post of the research assistant to any research laboratory. So that was our the contributions, or the contributions of the Dr. Gurude, and the number of specializations has been here into this particular department in the college, starting from the soil fungi. The specialization is there. The specializations of the mycorrhizal diversity, and the fungal taxonomy, and the endophytic fungi, the AMF, that is arbuscular mycorrhizal taxonomy, and the biotechnology, and the microfungal diversity. And then all the persons, the Dr. Hon'ble is there, who is working on the endophytes. Dr. Hedao is there, who is working on the macrofungal diversity. Dr. Magirwar is there, who is working on the your mycorrhizal technology. And the number of peoples are engaged here, though this is the degree college. We have a lot of work. All these peoples, I have seen that all these peoples are engaged for the, I don't say 24 hours, but at least for the 10 hours of a day. And they are contributing whatever they can carry out. They are contributing for the research purpose. And here, with this, I say to all the students that don't get afraid yourself. Because when I started my research, that time, I don't know anything about the mycorrhiza. I don't know anything. And I have started that work in 1994 or before that one. Just I have listened one lecture of Dr. Shri from the Jodhpur University, and he had given just a preliminary lecture. And I got interested, and at that time I wanted to start the research in that particular field, but I don't have any book, nothing. Even the microscope was not there. So even in that condition, I faced all the situations, all the difficulties. And I had been to Professor Ali from the Raipur. I had been to the C. Manoharachari to the Usmania University, Hyderabad. And I had been to the Dr. Bhagyaraj for getting me some training on the mycorrhiza. And after getting that training, I have started my research. The number of difficulty has come, but after all, I surpassed all the difficulties and started the research. So I say the students that don't get afraid. Start something. Because what happened? This is the age of the mobile. Everybody, you just listen yourself how much hours you are engaged on the mobile. So if you try to cut down that particular time, and if you focus on some other related things, then I can definitely say that you will get success, what you aim. So the aim is the very important thing. Your determination is very important thing. And you must think. What think? Not about the money, but you should think that how I can contribute for the nation. And if you can do that, then that will be in the real sense, that will be the Shraddhanjali to our great national leaders, Mahatma Gandhi. So don't spare much more time on the mobile. Spare it on the microscope. And I will say that the student must have the folded microscope costing ₹300 only. Keep it with you. And when you go into the field, just see what is there unique or what is different into the nature. That particular microscope, it is in their pocket. You take it out and see immediately on the spot. If you go outside of the college, and if you have the intention, then definitely you will get something new. And your name will be there encarved in the history of the mycology. And in the real cell, you will be the contributor of the nation. So with this, I thank you very much to the organizer for giving me this opportunity to talk and to express my views on this occasion. Thank you very much.
Thank you, Professor Devtare, sir. It's very nice to see after a very long gap of time. Sir, there was one sentence I would like to again share it. Spend your time on the microscope rather than mobile. And one of the participant, Bhargavi, has mentioned in this comment that she has liked this particular lines. So, dear participants, I would like to say that listening to Dr. Devtare is a feast of knowledge, and you have witnessed how what the way he expresses his thoughts, and most of the thoughts are based on their practical applications rather than their theory, idealism. Rather than idealism, it is based on the practical approaches. And sir, thank you very much for enlightening us. Now it's a time for the presentation of the different types of the events taken by our collaborator institutes. So there is a compilation of the video, and I would like to invite Swati madam to continue this program.
I compliment the organizers, the coordinators of the celebration of Fungal Awareness Week efforts that they have put in for this event, and for the participants that they have gained experience and knowledge. Now we will see the various activities performed by the various collaborating institute in the form of compiled video. So I request to sir kindly play the video. Activity.
Myself, Dr. Narendra Shahare, Professor and Head Department of Botany, Bridgelal Biani Science College, Amravati, welcome you all. In response to Professor Lean Body of UK, Mycological Society of India and Shri Shivaji Science College, Amravati, in the collaboration of these two institutes, we, the Department of Botany, Bridgelal Biani Science College, Amravati, celebrated the Fungus Awareness Week in our college on the 29th September 2021 by conducting the program Mushroom in Kitchen, Recipes and Preparation.
Good morning. Today on 1st October 21, Sri Shivaji Science and Arts College Chikli organized valedictory function of Fungal Rangoli competition. All over India, there is celebration of World Fungus Day 2021 on 2nd October 2021. Fungal Awareness Week from 20th September to 2nd October 2021 organized by Shri Shivaji Education Society, Amravati, Shri Shivaji Science College and Mycological Society of India. We organize the Fungal Rangoli competitions. 12 different types of events organized by various colleges. In this week, our college organizes Fungal Rangoli competition. A huge response for this competition. 45 responses are for this Rangoli competition. The Rangolis drawn by participants are very attractive, as well as they labeled with the scientific name of fungus. I congratulate to all participants who participates in this Rangoli competition. Professor Siddharth Mende, Assistant Professor of Department Microbiology, and Professor Mukund Kolte, Assistant Professor in Department of Botany, work as jury member for this Fungal Rangoli competition. I request to our principal, Honorable Dr. Omraj Deshmukh sir, for declaration of the today's Fungal Rangoli competition.
Thank you, madam. Good morning, all. I take opportunity to declare the result of Fungal Rangoli competition organized by our college, Shri Shivaji Science and Arts College, Chikli, in collaboration with Shri Shivaji Science College, Amravati, and Mycological Society of India. Congratulations to all participants. Our jury member selected best 3 fungal rangoli for awarding prizes. The third prize goes to Priti Hanuman Dahake, Shri Shivaji Science and Arts College, Chikli. Second prize goes to Ankusha P. Kochar, Bijral Beni Science College, Amravati. And the first prize goes to Kartiki P. Kaule, ICAR, CCRI, Amravati Road, Nagpur. I congratulate all the student participants for winning the prizes. Thank you.
Good morning and welcome to all of you. This is Department of Botany, Shri Shivaji Arts, Commerce and Science College, Mutala, District Buldana. Myself, Dr. Praveen Ajab Singh Theng, Assistant Professor and Head, Department of Botany. On the occasion of World Fungus Day, we organize essay competition entitled Fungi for Industries. For this competition, we have obtained overwhelming responses throughout India. I express my sincere gratitude to Shri Shivaji Science College, Amravati and Mycological Society of India to giving opportunity to organize such wonderful event. As a head Department of Botany, my best wishes to all participants
of human life. It helps the human in various aspects from food to various industries. For awareness and importance of fungi, we organize the essay competition on topic Society of India and Professor of Department of Botany, Shri Shivaji Science College, Amravati, for his constant inspiring approach and to give us such a great opportunity.
And PG student without registration fees on the topic Fungi for Industry. And for this competition, only scan copy in PDF format of neat and clean handwritten essay on page were accepted. So we got overwhelming result toward the research on applied mycology. Finally, I assure that we fulfill the aims and objective of this event. Thank you.
I, Dr. Suchita Khudke, Head Department of Botany, Vinayak Vignan Mahavidyalaya, Naggaon, Khandeshwar. As we know that fungi play important role in many aspects of human life, including medicine, food, and agriculture. These are the decomposers in most of the ecosystem. So this is the right time to make aware the world about this immense importance of fungi and mycology for the sustainable development. On the occasion of Fungal Awareness Week, our college, Vinayak Vignan Mahavidyalaya, in collaboration with MSI and Shivaji Science College, Amravati, has organized national level drawing competition on the theme Draw Your Favorite Fungus on 20th September 2021. Our college is the only science and commerce college which is affiliated to the Sant Gadge Baba Amravati University, Amravati. Our college always encourages students for the participation in various activities to develop the holistic approach. The aim of this competition was to develop the creativity in the student and to make them aware about the world of fungi. Thank you.
Hello, this is Anand Hivrade, Assistant Professor in Department of Botany at Vinayak Vignan Mahavidyalaya, Nandgaon, Khandeshwar District, Amravati, Maharashtra. On the occasion of celebration of World Fungus Day, our Department of Botany and IQC has organized national level drawing competition under the theme Draw Your Favorite Fungus on 28th September 2021. This competition was open for all the students of UG and PG. We got huge responses from all over the India, including the different states like Maharashtra, Karnataka, Kerala, Tamil Nadu, Chhattisgarh, Haryana, West Bengal, Tripura, Agartala, etc. We got total 329 drawings, in which 230 drawings from UG category and 99 drawings from the PG category. The drawings were judged on the basis of relevance of subject, creativity of Dr. Kishore Suradkar. I am very happy to be a part of this.
In collaboration with Shivaji Science College, Amravati and Mycological Society of India. As we know that the fungi are everywhere in nature, they are causing various serious diseases to the human beings and the plants, knowingly, unknowingly. Fungi can damage the human organs. The recent example is mucormycosis, and caused by the fungi known as mucor, that is black fungi. Therefore, there is a necessity to aware the student. Thank you.
I am very happy to, as a coordinator of Mycological biography competition. This is a collaborative work of Shri Shivaji Science College and Mycological Society of India. Nowadays, there are two sides of the coin. As such, the fungi has two sides. It is we are trying to familiar with the society as well as the UG. 70 students of the various universities, various colleges of the all India are participated, and 3 jury members selected the best biography of the various.
It is a very unique type of the activity which is conducted by SPM Gilani College, Ghatanji, and the responses are recorded, very good type of the responses, and I would like to congratulate. Sir, I would like to express some views about the world fungi.
Very good morning to one and all. I, Dr. Ankit Kare, Department of Botany, JD Patil Sangudkar Mahavidyalay, Daryapur College. I personally welcome you all on behalf of my college. Before presenting the brief report of EQ's competition, I would like to congratulate MSI, Mycological Society of India, and Shri Shivaji Science College, Amravati, and all other collaborators for successfully conducting the program. I hope in future also such activities will be performed and taken again for the beneficial of the students and the social persons, local persons. So here is the report of the e-quiz competition. So e-quiz competition on the topic fungal systematics and application was organized by Department of Botany and IQAC, JD Patil Sangudkar Mahavidyalay, Daryapur, District Amravati, in collaboration with Shri Shivaji Science College and Mycological Society of India. Quiz competition was organized on the occasion of World Fungus. Participation was not only from Maharashtra but also from other 11 states. These 11 states include Uttarakhand, Karnataka, Rajasthan, Madhya Pradesh, Chhattisgarh, Kerala, Tamil Nadu, Gujarat, Goa, Uttar Pradesh, Telangana, etc. I am happy, sir. The technical support was moderator of the program, and I again personally thank to the conductors.
On 30th September 2020, our botany department organized national virtual seminar competition based on theme fungal pathogens of human, animal and plants in botany department. The basic aim to celebrate this program is to make students aware about the importance of fungi in agriculture, medical field and its application in pharmaceutical industry, and also to create interest among the students. Our objectives is that after going through this topic, students will be able to understand awareness of how to prevent and head Department of Botany Winner Fungus Day with the students. Program was conducted smoothly by Mr. N.B. Chaukhande, and vote of thanks was given, made it a grand success. Thank you very much.
In the polythene bag, and on the polythene bag we are to write the name of the culture, name of the manufacturer and date of the manufacturer. So here all these things. Stop This is the Mahatrico that is available in market.
Hello everyone, myself Dr. Kishore Suladkar, Assistant Professor and Head Department of Botany, Indira Mahavidyalay, Kalam District, Yavatmal, Maharashtra. As we all know that we are celebrating the Fungal Awareness Week 2021 on the occasion of World Fungus Day. Collaboratively with the Mycological Society of India, Shri Shivaji Science College, Amravati, and other institute of student and a research scholar. We had received more than 250 participants for this competition from the various renowned institute and the university of the country, like undergraduate student, postgraduate student and research scholar. These three winners from each category will be felicitated in the valedictory function, and e-certificate will be provided to all the
Hello, dear participants. Now it's time to announce the winners of various competitions. Friends, about 12 institutes have participated in this event, and 3 winners from each college have been declared by the authorities. So I request Dr. Prashant Deshmukh to present the video of the winners announcement. Kindly bear with us. Dear participants, Brijlal Biani Science College, Amravati. Okay, the first Brijlal Biani Science College, Amravati. Mushroom in kitchen recipe and preparation. The first winner is Abhinav Kadu, Sipna Engineering and Technology, Amravati. Second, Kumari Netra Anasane. Third, Kumari Shreya Chopkar. Congratulations. Shri Shivaji College of Arts and Commerce, Akola. Under UG category, Mr. Jayesh A. Lagad. PG category, Kumari Firdos Fatima Mirza. Khalid Beg. NES Science College, Nanded. UG category, Kumari Rutuja Avinash Nihatkar. Rajashi Shaus Science College, Chandur. PG category, Kumari Roshni Gokul Prasad, Dhote Bandhu Science College, Gondia. UG category, Chitrali Kiran Sarunke, Modern College of Arts Commerce, Pune. PG category Komal Dinesh Srinivas, Sri Shivaji Science and Arts College, Akola. Next. Okay. Sri Shivaji Science College, Motara District, Buldhana. UG category, Tabassum A. Jamal Mohammed College, Trichy. Bharati Southern University. PG category, R. Nirmala Bhargavi, St. Joseph College, Autonomous College, Bangalore. UG category, Miss Nikita Anil Band for second number, Shivaji Agriculture College, Amravati. Under PG category, Ismat Sifras, Vivekananda Arts and Science College of Women, Sankari Periyar University. And third number under UG category, Rakshita N. Lakshmi, Annamali College of Women's, Autonomous, Bangalore. PG category, Hitenshini T.Y., St. Joseph College of Autonomous, Bangalore. Sri Shivaji Science College, Chikli. National Fungal Rangoli Competition. First number is Kumari Karthike P. Kavle, ICR CCRI Amravati Road, Nagpur. Second number, Miss Anushka K. Co-Chair, Brijlal Biani Science College, Amravati. Third number, Kumari Preeti H. Dhake, Sri Shivaji Science and Arts College, Chikli, Buldhana. Then another is e-quiz competition organized by JD Patil Sangrudkar Mahavidyalay, Daryapur. First number, Kumari Chabi Khemuka, Maharshi Junior College, Amravati. Second number, Miss Rinku Deepak Veer, Sri Shivaji Science College, Amravati. Third, Kumari Rinku Manoj Bansod. E-quiz on e-photography competition under UG category. First number, Pratik V. Zumbre, College of Agriculture, Dharwa. PG category, Rashmi Ishwar Rao Chandan Khede, MAK College, Hinganghat. Under UG category, second number, Ratrika Roy, NES Ratnam College of Arts and Science. Under PG category, Prathamesh Pramod Mopalwar, Mohta Science College, Nagpur. Third, UG category, Pratik Zumre, College of Agriculture, Dharwa. Under PG category, Priyanka Surarkar. And under research category, Vishal Narayan Taide. Next. Okay, mycological biography competition. ठीक है. Mycological biography competition. The first winner, Kumari Nikita Anil Band, Sri Shivaji Science College, Amravati. Second number, Miss Prabha, DSB campus Nainital. Third number, Kumari Rutuja Niharkar, Rajarsi Shahu Mahavidyalay, Chandur Railway. Okay, national level drawing competition. The first number goes to UG category Kumari Dipantu Datta, Holy Cross College, Agartala. Under PG category Kumari Prachi Nipase, Sri Shivaji Science College, Amravati. Second number UG category Kumari Jayashri Tankar, Vinayak Mahavidyalay, Nandgaon, Khandeshwar. PG category Kumari Kartiki Kavle. UG category third number Pinchan Iftekhar, Maharashtra Udaygiri Mahavidyalay, Udgir. PG category Kumari Shital Marbate, DB Science College, Gondia, third number. Next. Page this. Yes. I request Dr. D.V. Hande sir kindly announce the names.
National Photography Competition on fungal diseases of fruit and vegetable. UG category Kumari Baharuddin Fatima, Jamal Mohammed College, Tiruchirappalli. PG category Sonali Gaiki, MSc first year Vidya Bharati Mahavidyalaya, Amravati. Research category Kumari Nivedita Manogaram, Madras Christian College, Tamil Nadu. UG category Kumari Sanika Ahir, Sri Shivaji College, Akola. PG category Kumari Akshada Srikande, MSc first year Shivaji Science College, Amravati. And the research category Mr. Thirucharan Narayan, Banaras Hindu University, UP. Next. National Online E-Poster Competition from UG category Kumari Utkarsha Vilas Wankhede, Vidya Bharati Mahavidyalaya, Amravati. PG category Kanifa Burgate, Parbani. From research category Jain Vinod Chandra Patel. Next.
Okay, thank you very much, Professor Dilip. Friends, okay, we will share the PDF of this result on our WhatsApp group. Friends, it's a pleasure to invite a very renowned and esteemed guest, Dr. Munruchi Kaur, the Vice President of MSI, for her remark as a chief guest. Friends, Dr. Munruchi Kaur is a professor and head of the Department of Botany at Patiala University. Second, she is also a director of UGC HRDC at Punjabi University, Patiala. Dr. Munruchi Kaur has completed his MSc from the Patiala University and she secured a gold medal. She also completed her MPhil PhD from the Punjabi University. She was also awarded with SK Shom Memorial Lecture. And presently, she is also Vice President of MSI. Dr. Munruchi Kaur has more than 24 years of teaching and research experience. She has to her credit 69 research papers published in national and international journals of reputation. She has contributed 9 book chapters. Her research in the field of mushroom diversity and systematics speaks volumes of her expertise in the lamellate fungi. While working in the field of taxonomy of lamellate mushrooms in the northwest region in India, she has her credit 38 species reported of 5 genera and 194 species as a first time record from the India. Along with looking Frogeri's diversities, the focus also was on looking for the wild edible agaricus from the northwestern regions of the India, with the aim to domesticate these agaricals, the edibility status based on the mycological data collected from the local people. So friends, I would like to invite chief guest Professor Munruchi Kaur to deliver his chief guest address.
Thank you for that introduction, sir. Wishing you all a very happy Gandhi Jayanti. Respected mycophiles, as we all are, a very good afternoon. On the onset, I would like to acknowledge Shivaji Education Society, Amravati, for nurturing these prestigious group of institutes. Professor G.N. Chaudhary, Principal, Shivaji Science College, Amravati, Professor B.K. Dorkar, Head Department of Botany, Shivaji Science College, Amravati, Organizing Secretary, the ever energetic Professor D.V. Hande, then Joint Secretary and co-conveners, the faculty of the participating colleges, as well as Shivaji Science College. Then Professor Rodrigues, President MSI, Professor Raman, Secretary MSI, and the senior mycologists on the dais. I understand the type of efforts that have been put in to celebrate this World Fungus Day. It is not easy to organize such big events, and I congratulate the office bearers and the organizing secretary for putting in these efforts and popularizing these fungi. Plants and animals, as we all know, are the visible world, while fungi are the invisible world, but very important in their role. These tiny little organisms, they are responsible for scavenging of the litter from the planet Earth. The fungus world encompasses an enormous diversity of taxa. They are varied in their habitat, their life cycle, and in their importance to mankind. They serve as food in the form of mushrooms, truffles. They are the fermenters. They are giving us very important, industrially important enzymes. Then they are also the biological pesticides to control the pests and weeds. Then they produce a number of bioactive products, psychotropic compounds, which are a very hot area of research in fungi these days, the magic mushrooms. Then they occur as symbionts in the form of mycorrhiza. Much of the research is on the fungi in this fungi world. Then taking into consideration the enormous activities that are attributed to the fungi, although all the mycologists that are there, we are well aware that we are very passionate about our researches in the fungal world. It is the duty that is to be shouldered by us to make this invisible fungus visible to the world and to ignite the young minds to take up the researches in the field of mycology and plant pathology. And for taking up this responsibility, I think the Amravati Science College has taken up a great opportunity by celebrating this World Fungus Day, dedicating a full week to highlight the importance and recognizing the fungal world by organizing varied programs in the form of lectures. Then by organizing photographic competitions on the fungal world of fungi, especially the photography competition to do with the diseases of fruits and vegetables. Then they organized essay competitions on the fungi for industry, drawing competition with the slogan that draw your favorite fungus. Then the poster competitions on fungi, boon to mankind. The e-quiz idea of was conducted on the systematics and applications of these fungi. The competition on mushroom kitchen and the rangoli must have been one of the most popular ones and would have been a great fun. It is indeed, a very apt way of popularizing the fungi. And it is in the true sense, the celebration of the World Fungus Day comes to make sense at this national level in our country. I put on record that it is the first ever attempt of celebrating the World Fungus Day in India, and it will go down in the history of MSI that it was Amravati who took the lead in the celebration of the World Fungus Day at the national level in our country. I congratulate all the winners of the various competitions, and I am sure that all the events, they were indirectly igniting the young minds to be taking interest in the world of fungi. And I hope that the efforts that have been put in by the team under the secretaryship of Dilip Hande and under the aegis of Mycological Society of India will be fruitful, and in the future these young minds would be ignited enough and they would be passionate about fungal research, and a number of students there will be allured to take up mycology and plant pathology as their profession. I congratulate the Mycological Society of India. I congratulate all the participating institutes under the aegis of this Shivaji Science College. I congratulate each one of you. And in the end, I acknowledge the efforts put in by all of you. And I bow my hat to my mentor, Dr. N.S. Atri, for mentoring me into this world of mushrooms. Thank you, one and all.
Thank you. Thank you very much, Professor Munruchi Kaur, madam. It's a very nice to see Professor Atri sir, you again after a very long gap of our MSI conference, and we are really delighted to be a part of this event. Now I would like to call upon Dr. Swati madam for the vote of thanks.
Gratitude is the fairest blossom which springs from the soul. Every celebration is an experience. We remind that events like this one are a milestone for our department and college also. It's time to acknowledge the known and unknown helping hands who have efforts to make this event successful. May I call upon Dr. D.G. Hedau sir for the vote of thanks.
Good afternoon, everybody. Honorable Chairman of World Fungus Day, Valedictory Function and Winner Declaration Program, the Chief Guest, Office Bearers of MSI, Senior Mycologists, Honorable Chairmen of the various sessions, guest speakers, professors, teachers, research scholars, and dear students of the different parts of the world. In the capacity of co-convener of this national level fungal awareness week and celebration of World Fungus Day, organized by the Department of Botany, Shri Shivaji Science College, Amravati, in association with prestigious Mycological Society of India, I feel proud and privileged to propose the vote of thanks. At the outset, we express our gratitude to respected Professor B.F. Rodrigues, President MSI, and Professor N. Raman, Secretary of MSI. Also, the executive members for the alliance with our college to organize this unique event in a great and successful way. We are highly grateful to respected Professor T. N. Lakhanpal, former president of MSI and well-known mushroom man of India, for being chief guest and inaugurator of Inaugural Ceremony. His experienced speech is always a source of inspiration for us. We are very fortunate to have a very good researcher and a charming personality in the field of mycology, Professor B.F. Rodrigues. We thank him for his presidential remarks. The words of gratitude definitely will not enough to acknowledge to our beloved principal, Professor G.N. Chaudhary, for his presidential address. Organizing committee is wholeheartedly thankful to Professor B.F. Rodrigues for his very informative talk on mycorrhizal fungi. Organizing committee extends our thanks to Professor Rupam Kapoor, former president of MSI, Department of Botany, University of Delhi, for her very enlightening speech on Botrytis cinerea. Technical session 1 was chaired by well-known environmental microbiologist Professor D.J. Bhagyaraj, former president of MSI, whose contribution in mycorrhizae is noteworthy. This session was co-chaired by Professor Naveen Congo, Professor and Head Department of Microbiology, Dr. Harisingh Gaur, University, Sagar. Dear participants, technical session 2 was chaired by renowned researcher in the field of mushroom biology, Professor N.S. Atri, former president and editor-in-chief of Kavaka, Department of Botany, Punjab University, Patiala. This session was co-chaired by our friend Dr. Aniket Gade, young scientist awardee and researcher in the field of nanotechnology, Department of Biotechnology, HGB, Amravati University, Amravati. Let me record our deep sense of gratitude towards all the chairman and the co-chairmen who have kindly consented to chair the sessions and expressing their remarks during this event. The event would have been incomplete without the presence of respected Professor P.W. Devtare, our former head Department of Botany, Shri Shivaji Science College, Amravati. Sir, your presence has glorified this valedictory function. We are indebted to you for your valuable presidential address, sir. We are very fortunate to have a very good personality in the field of botany, Professor Munruchi Kaur, Vice President of MSI and Head Department of Botany, Punjab University, Patiala. Madam, we are highly grateful to you for your guidance and presence as a chief guest. Let me acknowledge all the hosts and the co-hosts of various sessions for smooth conduction of this event. Dear friends, to organize such a mega event was not possible without the collaboration and the active support by our 12 collaborative institutes, namely Brijlal Biani Science College, Amravati, Shri Shivaji Science College of Arts, Commerce and the Science College, Akola, Vinayak Vidyan Mahavidyalay, Nandgaon, Khandeshwar, Rajarshi Shahu Science College, Chandur Railway, SPM Science and the Gilani Arts Commerce College, Ghatanji, JD Science Patil Sanglutkar Mahavidyalay, Daryapur, Indira Mahavidyalay, Kalam, Shri Shivaji Arts Commerce and Science College, Motala, Bapurauji Butle Arts, Narayan Rao Bhatt Commerce and the Bapu Saheb Patil Science College, Digras, the Science College of Agriculture, Biotechnology, Amravati, Shri Shivaji Arts and the Science College, Chikli, and the last, the Government Arts and the Commerce Girls College, Raipur. Organizing committee is humbly thankful to respected principals and coordinators of all the colleges. To complete the entire editorial work of the souvenir within a very short period was the challenging task before us. Being the convener of editorial committee, I am proud to say that the entire work has been completed with the great efforts taken by my committee members. Thanks to all. I'm also thankful to all the authors who have contributed their valuable work in this souvenir. We must acknowledge the charitable publicity offered by papers and the electronic media to draw attention of the common citizen of this region towards such magnificent event organized by our department. We are grateful especially to the members of the local advisory committee and MSI members of Amravati region. I heartily congratulate to the winners of all the participants for their active participation in various activities undertaken. I thank to our distinguished guests, Fungus Day will guide us on many counts. Dear participants, we hope to have your enormous response in future as well as for such academic ventures. Thank you. Thank you very much.
Thank you very much, Ganesh. Ladies and gentlemen, this event would be possible with the generous support and appreciation from the Mycological Society of India. And we are grateful to all MSI office bearers for giving us this opportunity to share this platform to meet the brilliant minds of our country. Now I invite the Secretary of MSI, Professor Raman, to express a vote of thanks and share his outlooks. Professor Raman.
Am I audible?
Yes, yes, sir.
Yes. Friends, in the annals of Mycological Society of India, this is a wonderful day. We conducted the World Fungus Day 2021. So just like Gandhi Jayanti, this day will be World Fungus Day Jayanti year after. So at the outset, I thank Shri Shivaji Educational Trustee for their fullest support with the Mycological Society of India for this event to happen. I thank Sri Shivaji Science College, Amravati, and all the other 13 colleges who have contributed a lot to conduct this function. On behalf of the Mycological Society of India, I thank the Principal, Shivaji College of Science, Amravati, the HOD, Department of Botany, and the former HOD of Botany, Professor Devadare, and all the team. So as Professor C.B. Subramaniam told, togetherness achieved this reward. I can say it is a reward by the teamwork. I congratulate, I thank Professor Ande, Dilip Ande, for coordinating this one and made this function to happen, the World Fungus Day. On behalf of Mycological Society and on behalf of all of you, I thank Professor Lynn Bodie, the British Mycological Society, for giving the opportunity to celebrate this one. And I thank all the office bearers of MSI and all the members of MSI, all the participants of this event, and all the chairperson and the speakers of this event. for the fullest cooperation they extended in the annals of the Shri Shivaji Science College, Amravati. This day will be again a wonderful day. Again a wonderful day. The Mycological Society of India is approved the initiation of MSI Amravati unit to Shivaji College, Shri Science College, Science Shivaji College, Amravati. My heartiest congratulations on behalf of the MSI and all the office bearers, all the MSI members. This is a wonderful day again for the Shri Shivaji College and particularly for Professor Hande, Dilip Hande. Congratulations and thank you very much.
Thank you, sir. Thank you very much.
Thank you very much, sir. It's a really wonderful day for us also. And I do have an opportunity to meet Professor Raman in MSI conferences. And I personally found very enthusiastic, supportive, and appreciating all of us, the people who are coming across the regions. And in MSI conferences, he is also one of the center of attraction. And as T.S. Lakhanpal sir said in his speech, he is one thread. He is the one that particular thread who bonded us with love and affection in these academic endeavors. Thank you, sir, for showing trust on us and hope you will provide opportunity to host such programs in future also. Now, I appeal everyone to dear friends, MSI unit Amravati is approved by our Honorable Professor N. Raman. So we all the members of Shivaji Science College really grateful for that, and thank you very much, sir. Now I appeal everyone to kindly stand up for the national anthem.
Punjab, Sindh, Gujarat, Maratha, Dravid, Uchal, Banga, Himachal, Yamuna, Ganga, Uchala, Jala, Siddharanga, Jabasudasi Samagi, Jaya Jaba Jaya Gada, Jana Jana Mangala Jaya Jaya Bharat Bhakya Vidada, Jaya He, Jaya He, Jaya He.
Okay, with the permission of this chair, this session is concluded.
Yes.
I'd like to call upon Dr. Dilip Hande to say a few words.
Thank you, all the senior mycologists, president, secretary, and office bearer of Mycological Society of India for showing the faith on our capabilities and approved as a MSI Amravati unit. This is a great achievement. Thank you very much.